Welcome to the 12th EAI International Conference on Bio-Inspired Information and Communications Technologies. I'm Professor Yifan Chen from University of Electronic Science and Technology of China, and I'm the General Chair of this conference. So there are three main topics of BICT 2020. The first topic is about direct bio-inspiration, which includes systems utilizing biological materials, processes, and systems, such as molecular communication and computing, DNA computing and memory, brain-machine interface, and synthetic biology. The second main topic is about indirect bio-inspiration, which include systems and application design upon biological principle, processes and mechanisms such as neural networks, evolutionary computation, swarm intelligence, amorphous computing, and cellular automata. The third main topic is about foundational bio-inspiration, which include biology and ICT evolving similar schemes and validity, such as evolution, game theory, network theory, measures of validity and validation of methodology. The conference starts from year 2006 in its original name as Bionetics. So from 2006 and 2012, the conference is called Bionetics. And since 2014, the conference has been renamed into BICT. So the past few editions of BICT were held in Boston, New York, Hoboken, and Pittsburgh. The organizing committee members include myself as the general chair. The TPC chairs include Prof. Tadashi Nakano from Osaka University, Prof. Lin Lin from Tongji University, Prof. Wei Siguo from Warwick University, and Prof. Muhammad Mahfouz from University of Wisconsin, Green Bay. The publicity and social media chairs are Prof. William Casey from Carnegie Mellon University and Prof. Andreani from Stevens Institute of Technology. The publications chair is Prof. Qian Liu from University of Electronic Science and Technology of China, and the workshops chair is Dr. Yotaka Oke from Osaka University. The tutorials chair is Dr. Peng He from Chongqing University of Post and Telecom, and the sponsorship and exhibits chair is Dr. Hui Liu from University of Science and Technology of China. Our local chair is Prof. Hao Yan from Shanghai Jiao Tong University, and the web chair is Dr. Yue Sun from Chengdu University of Technology. Conference manager is Carolina Masinova from EAI. Now, overall, we have 30 full paper submissions and 27 short paper submissions. So in total, there are 57 submissions. Each paper is reviewed by 2 to 3 TBC members. And out of all the papers submitted, 24 papers have been accepted and 8 short papers have been accepted. So the acceptance rate is 49%. Now there are 2 keynote speakers. The first keynote speaker is Prof. Andreani Odysseos from University of Cyprus. Her topic is on bio-nanomachine diagnostics and nanonetwork therapeutics in brain malignancies, emerging transdisciplinary approaches with bio-nano device interfaces. And another keynote speaker is Prof. Pedro Antonio Valdez Sosa from Cuban Neuroscience Center. His keynote speech topic is global brain projects and global health. So authors of selected papers will be invited to submit an extended version to MONET, Mobile Networks and Applications, IEEE Trends on Nanobioscience, IEEE Trends of Molecular, Biological, and Multiscale Communications. And here is the highlight of technical program. For day 1, we have keynote speaker Prof. Andreani Odysseos from Cyprus University. And day 1 also includes 1 technical section and 3 special sessions, which include data-driven intelligent modeling application and optimization, intelligent Internet of Things and network applications, and intelligent sensor networks. In day 2, we have another keynote speaker, Prof. Pedro from Cuban Neuroscience Center. So day 2 also includes 1 technical session and 1 special session on Internet of Everything, and 1 workshop on applications, testbeds, and simulation designs for molecular communication. So thank you very much for attending BICT 2020.
Dear ladies and gentlemen, let me welcome you to EAI BICT 2020 conference held in this virtual interactive setting. I am Karolina Marcinova, the EAI conference manager for BICT 2020. Unfortunately, due to the virus, I am not able to meet all of you in person, so I'm using this opportunity to address the organizing committee, the keynote speakers, the authors, and the participants on behalf of EAI. I would like to thank all of you for being a part of this conference and for your involvement with EAI. In particular, I would like to thank the General Chair Yi-Fan Chen and the Program Chair Tadashi Nakano, together with the whole organizing committee, for their hard and excellent work throughout the whole process of conference preparation. Today, during the streaming, you can actively participate in two ways. Firstly, you can join the Q&A on Slack through a link you can see below this video. Upon accessing the EAI BICT 2020 Slack workspace, you can enter all the channels which are divided by sessions. Secondly, you can vote on individual presentations and leave your fellow presenters feedback on their work through EAI Compass. We will shortly show you how to access this platform and how to leave and receive feedback for presentations in this conference. I would like to also use this message to invite you to join us again at EAI BICT 2021 that is planned to take place in Osaka, Japan in August 2021. Should you be interested in becoming a part of next year's organizing committee or the technical program committee, please do not hesitate to contact me at my email address below. Similarly, if you are interested in discussing other possible cooperation, a conference, a workshop, or a new event altogether, please contact me at my email address as well. Finally, the brochures for EAI BICT 2020 are available on the conference website. In there, you can find further information about the keynote speakers, the organizing committee, the technical program committee, EAI's platforms and activities, and much, much more. Now my colleague Michal will present you with what EAI does, who we are, and how you can get involved in our various activities. Thank you for your attention and enjoy EAI BICT 2020.
Hi everyone, my name is Michal Dudic. I'm the community manager at EAI, European Alliance for Innovation. It's my pleasure to welcome you at this conference and say a few words about who we are and what we can do for you and your research career. In short, EAI is a global community for a greener, healthier and smarter world. As of today, we are home to more than 60,000 members from 167 countries, and we reach out to tens of thousands of subscribers. As an organization, we are non-profit from day one, and what is most important to us is that we remain open to all researchers from all around the world thanks to membership that is completely free. We organize more than 100 events annually, such as this conference, and we do so in publishing partnership with Springer. I said in the beginning that EAI is a community, so let's talk about what that means and what it means for you. To put it briefly, we give our members a platform that builds their research. We do it with 3 main online community services where members come together to help each other write a better paper, get an objective review, and get recognized fairly. The 3 services in question are EAI Compass, Community Review, and EAI Index. Firstly, EAI Compass is an online app where you can meet and connect with new colleagues and get feedback on your paper as well as your presentation. In addition to that, it lets you download all full papers that will be presented at this conference, and you can vote on your favorite presentations as well as see everyone who is here and connect with them. You can do this right now if you go to EAI Compass website, compass.eai.eu. Next, we are improving the classic conference review process with community review. It has already been in use at all our events since 2019, and we were very excited to hear a lot of positive feedback from program committee members regarding the reliability and the speed of the community review. Let's talk briefly about what community review does. Essentially, it is a website that shows abstracts of papers that are right in the middle of the review process, as long as the authors allow it, of course. And all EAI members may then bid to review specific papers. When they submit their bid, they put in their bio and their qualifications, which are sent to the program committee, who can then decide whether or not this bidder is qualified to review the paper they bid on. This relatively easy access to review opportunities means that bidders really need to put their best foot forward if they wish to be selected, which improves the quality of the entire review process. At the end of the day, this benefits you, the author. And last but not least, let me tell you a thing or two about EAI Index. EAI Index is our credit-based evaluation system that we rolled out this year to all of our conferences and journals that allow you to climb the global ranks of EAI community and get recognized for your work. It calculates a number value for most actions you make, such as getting your paper accepted or submitting a review, and these numbers accumulate for 12 months. At the end of this 12-month period, we put together a ladder of all EAI members, and the ones at the top receive a nomination to one of the membership ranks: senior member, distinguished member, or fellow. For each action that is eligible for EAI Index credits, we'll look at the quality of your action as it was evaluated by another member of the community, such as, for example, the review score of your submission. To make sure that the system is fair to newcomers, every 12 months the credit count gets erased, the ones at the top receive their nominations, and every member starts at 0 for the following 12 months. And finally, Smart Submit is a collaboration feature that is coming later this year. It will allow you to submit your research ideas and your work in progress abstracts to get the kind of help and feedback you're looking for. Maybe you are looking for co-authors, maybe you would like to find a mentor or a mentee, or maybe you want to find out how the community feels about your idea. This is what Smart Submit is designed for. Ultimately, it's about helping you write a better paper and increasing your chances of getting accepted. Again, we will be launching this feature later this year, so stay tuned. And so I'm going to leave you with many different ways to get engaged at different levels. There are lots of opportunities in many of our events and publications, which means many ways to connect with people and collaborate. You may learn more about everything I just talked about at our website, eai.eu. These services exist to help you and to make your lives easier, so we encourage you to send us your comments, ideas, and feedback to community@eai.eu. And if you're interested in volunteering and contributing, you can let us know at the same email address. Don't forget that you can use EAI Compass to vote on presentations in real time to determine which ones are the best, as well as to download all full papers that will be presented today. Just make sure that you log in using the same email address as the one you used to register to this conference. Thank you very much for your attention. Please enjoy the conference, and I hope we will see everyone online soon.
Greetings from Cyprus, everybody. It is indeed an extreme honor for me to deliver this keynote address, especially under the unprecedented conditions we are experiencing nowadays. I wholeheartedly thank the organizers for this unique opportunity. And what I shall try to share with you in the next 40 minutes or so is an overview of the latest advances in molecular communication, and in particular bio-nanomachines and nanonetworks under the control of interfaces emerging as diagnostic and therapeutic tools with emphasis on brain malignancies, the most detrimental human cancer. Some 150 years ago, Claude Bernard, a renowned French physiologist at the time, defined medicine as a fragile art applied on solid sciences. I literally cannot think of any better example than the implementation of molecular communication concepts and technologies in the management of brain malignancies, which are the epitome of complexity in human diseases. Nanotechnology, as a discipline, was born at the monumental lecture of Richard Feynman. There is plenty of room at the bottom, where he described atomic scale machines and the vision that these machines can realistically function simultaneously. However, he acknowledged that this cannot happen because we, the scientists, are too big. This was, in fact, the first promise for the transformational potential that nanotechnology was going to have in the years to come. The tiny machines would be nothing else but nature's biomolecules in a senseless. So nanobiotechnology and nanomedicine came to the fore with Isaac Asimov's fantastic voyage, where 4 men and 1 woman in a jet submarine navigated through a bloodstream of a man, targeting a clot in the brain. What seemed to be fiction science at the time was in fact the first conception of mobile bio-nanomachines. According to my Greek history teacher, if you do not know where you are coming from, you can never know where you're heading to. In order to identify current challenges in nanobiotechnology, we have to undertake a path through history. Early on, scientists were confronted with a series of challenges associated with the acknowledgement that this new technology was defined by 2 divergent systems. Biological systems, which are fundamentally wind and organic, whereas most nanofabricated systems are hydrophobic and made of inorganic materials. Therefore, actual progress in this field was made possible only when the initial seminal advances of each contributory field came to fruition. In early 1990s, the challenge was how to physically couple the 2 divergent systems, since biomolecule material interaction is the sine qua non in nanobiotechnology. Experiments were undertaken to specifically couple biology with nanofabricated devices and tools. In 2000s, the challenge was how to circumvent the divergent systems by implementing nanomedicine artificial bio-nanomachines, such as common nanoparticles. By early 2000s, scientists began investigating how to integrate artificial bio-nanomachines into nanonetworks and nanodevices. Being at the dawn of 2020s, the challenge we are now facing is to realize the great promise in disease diagnosis and therapy with nano-device control. It is said that history is repeated, and molecular communication do verify this. Now, 2 divergent systems define a new information and communication paradigm. As you all know very well, computers and networks are simple, fast, and tough. Whereas, and believe me, I can confirm this, biological materials and entities are complex, slow responsive, delicate, and self-determined. Actual progress in this field, molecular communication, began when conceptual similarity was identified between seemingly very different networks and biological communications, or bio-nanomachines. In early 2000s, the challenge was how to map biological communication key concepts, such as information sources, senders, receivers, information carriers, encoding, decoding, and information propagation in networks. By early 2000s, information theoretic work drives molecular communication to a scientific discipline. And shortly afterwards, significant results in physical layer issues were reported. The challenge at the dawn of 2020 is now to realize the great promise of Richard Feynman in disease diagnosis and therapy with bio-nanomachines and nanonetworks. The first milestone on this pathway has been the presentation of a complete molecular communication paradigm by the SUDA team at the University of California at Irvine in 2005, where senders and receivers communicate with chemical signals and molecules. In an excellent retrospective review, members of the molecular communication founding team summarize key milestones that have had notable impact on the evolution of the molecular communication field, both at the conceptual and experimental level. I would like to point out those with indirect impact on the work that I tend to share with you today. These include the proposition of the Internet of Nanothings in 2010, the first research with specific application in biomedicine, such as drug delivery in 2013, the development of the first external interfaces in 2014, and at the end of this exponential phase, two critical artificial cell milestones. Two critical milestones, the demonstration of the first experimental testbed with engineered artificial cells and the proposition of the Internet of Bio-Nanothings. The launch of FedCSA Circle, a concerted action funded by European Commission, which was the first molecular communication activity funded by European Commission, could be considered as an acknowledgement of the high potential of this field. There is absolutely no doubt that the epitome of convergence of nanobiotechnology with molecular communication resides in their application in the management of malignant tumors. In 2014, the Russia team reported a communication model between nanoparticles and engineered tumor in vivo, integrating nanomedicine, tissue engineering, and communication. It was shown that the signaling or sender module, which is here blue, broadcasts the tumor location to the receiving module, red, in the bloodstream for amplified tumor targeting. Only 2 years later, the Felicenti team reported an integrated model of drug delivery. An actuator nanomode is in charge of delivering drugs, which here is shown as red particles. A monitor or control mode is in charge of assessing the effect of drugs on the target by measuring specific tumor quantities, which in practice are disease biomarkers released by the tumor. Here are shown as green particles. Finally, controlling the emission rate of the drug release via feedback signals, shown here as blue particles. It was around the same time when the mostly revolutionary conceptual framework of the externally controllable molecular communication was presented, interfacing bio-nanomachines with the external environment and bringing new challenges to the scientific community. The first comprehensive response to the search of in-messaging and out-messaging interfaces was the application of optogenetics, which means gene regulation via optical signals, for localized drug delivery in the brain by the European Commission-funded project StarTest. In search of bio-nanomachine to bio-nanomachine interfaces has led to the identification of emerging roles for synthetic biology and nanobiotechnology, and new approaches in implantable cells research. Ultimately, the need to decipher molecular mechanisms deep in the human body, which is directly related to brain tumors, is leading to new approaches in wearable device research and promises to take brain-machine interfaces to a new level where computing systems could detect, localize, and control molecular activities. This very simple graph, which discloses output in molecular communication in terms of peer-reviewed publications, delivers some very interesting messages. After an exponential growth in the late 2000s up to mid-2010s, the field seems to have reached a plateau in the late 2010s, reflecting the need of keys that will unlock new breakthroughs. These include the balance between theoretical and empirical research, the need for applications and reaching out to wider disciplines, and the expansion and growth across disciplines such as physics, mathematics, and medicine. This deceleration of scientific outputs has also defined open research challenges, including the establishment of a standard architecture and simulation framework of molecular communication, the design of communication protocols for value-added higher layer functionalities, and of external interfaces to interconnect with the internal molecular communication environment, which is an enabling technology that helps realize the Internet of Bio-Nanothings. Also, the deciphering and endorsement of collective behavior of bio-nanomachines, and finally, learning from evolutionary and adaptive nature of biological systems, implementing biosimilar molecular communication, and of course, obtaining deep insights and understanding of biological processes. So if Richard Feynman was here with us today, he would certainly say that yes, there is plenty of room at the bottom for molecular communications. Extending the roadmap set by Nakano and Suda, we come across two milestones highlighting acknowledgement and rewarding of bio-nanomachine and nanonetwork research that herald therapeutic translation of models and concepts. The Nobel Prize in Chemistry in 2016 for the design and synthesis of molecular machines, and the launch of Gladiator Fed Open, an international transdisciplinary integrated clinically applicable approach of autonomous externally controllable nanonetworks with bio-nano device interfaces in 2019. Glioblastoma multiforme, the most detrimental human cancer, is widely recognized as a model of wireless bio-nanomachine communications. It can be characterized as the convergence of complexity, stochasticity, heterogeneity, dynamics, and multi-scale natures that together characterize molecular communication networks. For those of you not familiar with the molecular mechanism of cancer, a tumor starts when a multipotent cell we call cancer stem cell, and in this case we call glioma stem cell, which carries multiple mutations that can drive transformation to malignancy, start progressing. This progression can be either transition to or transformation to a new phenotype, new characteristics, of which collectively make the characteristics of cancer. And this function we call GO, or proliferation and self-renewal, maintaining the same multipotent phenotype in a function we call growth. Glioma stem cells are those cells which survive surgery, radiation therapy, and chemotherapy, and under certain conditions, they repopulate and form tumor recurrence. So cell-cell interaction and microenvironmental factors play important roles in regulating the phenotypic switching and maintenance of distinct subpopulations of cell bio-nanomachines as either GO phenotype or growth cells and recurrent tumors. So existing models of glioblastoma growth nowadays are based on the pattern of spreading at the population level, and therefore there is an urgent need of models enabling the study of how cell bio-nanomachines interact as they spread. To meet this compelling need, it is highly imperative that us, molecular oncologists, start talking wireless and start translating tumor evolution to the concepts of molecular communication. environmental cues in the tumor extracellular matrix, the matrix around the cells, present especially graded doses built in multiple modes of communication. The sensitivity to spatial gradients of solid chemical factors and ligands is known as chemotaxis. The sensitivity to gradients of physicochemical properties of cell adhesion substratum, very important in tumor spreading and metastasis, is known as haptotaxis and is based on graded surface of the surface-bound extracellular matrix products. Going a step further, topotaxis is the direct migration guided by the gradient of the nanoscale topographic pictures in the tumor's microenvironment. It reflects the outcome of the material properties of cells and their surrounding extracellular matrix. And ultimately, coming to more mechanical properties, Eurotaxis mode is the sensitivity to gradients of substratum, stiffness, and rigidity, denoting the presence of mechanochemical cues, and is associated with adhesion substrata. We therefore require models of recapitulating the three-dimensional topographic complexity at multiple scales of size and organization. In a recent joint undertaking with the Nagano group in Japan, we studied growing bio-nanomachine networks and their applications to glioblastoma evolution and progression within these modes of communication. We first developed a growing bio-nanomachine network model that captures the dynamics of glioblastoma stem cells. And here we present the simulation of the spatial distribution of proliferative bio-nanomachines presented by the black dot and invasive ones presented by the red dot. Two bio-nanomachine subpopulations with differential migrating potential form a growing networks. Aptotaxis and chemotaxis gradients of molecules result to attractive or repulsive forces. And proliferative bio-nanomachines stay closer together to form a primary cluster. And probabilistically, they switch to invasive bio-nanomachines and migrate radially from the primary cluster. Under certain conditions over time, invasive bio-nanomachines switch back to proliferative ones to form secondary clusters. So ultimately, proliferation and invasion labor is divided to form a large-scale network collectively. In a wet lab experiment of human glioblastoma cells in a two-dimensional cluster, we show that the theoretical and experimental models of glioblastoma networks, evolution and progression are greatly aligned. And this leads to the conclusion that targeting the molecular communication network and microenvironment can be high promise for future targeted therapies. Enjoy. You can see in basic time irregularly shaped and spheroid cells which do not follow attractive forces but only repulsive and aggregate in three-dimensional structures here at the center. Yes, the theoretical and experimental models of glioblastoma networks, evolution and progression are highly aligned. So targeting this molecular communication network gives very high promise for future therapies, and this is going to be our immediate next work. And I hope we have the opportunity to share this, this new work in the future. The complexity of chemotaxis and comotaxis gradient, as we described them before, as communication modes, are shown in the interaction between glioblastoma stem cells and another subpopulation of early glioblastoma oncogenesis, the so-called glioma endothelial cells, which form the lining of new vessels, functioning in a dynamic interplay in a network of mobile bio-nanomachines. Tumor vessels can be recruited from glioma stem cells, and at the same time, glioma endothelial cells have direct effect on distinct subpopulations of glioblastoma cells, orchestrating the activation of migratory, invasive, or self-renewing potential of these glioma stem cells. In turn, glioma stem cells give rise to functional endothelial cells, and in a complementary network, remote progenitors from bone marrow generate glioma endothelial cells. Now, disrupting this network can become a central therapeutic challenge in glioblastoma management. Glioma endothelial cells and glioma stem cells are organized into a unique structure, the so-called glioblastoma perivascular niche. The perivascular niche is of major clinical significance, as it is here where the tumor recurrence arises. In terms of bio-inspired information and communication technologies, this tumor structural unit is expected to attract great attention as a prototype leader follower based mobile molecular communication network. And this is because physical association and release of soluble factors via cell-cell interactions are augmented and amplified when interacting cells are in close proximity. In order to functionally maintain cell renewal, the glioma stem cells require several autocrine and paracrine factors that are released in the neighborhood of the perivascular niche. And the perivascular niche does allow access to increased localized concentration of these soluble factors secreted by glioma endothelial cells. Furthermore, the physical proximity of glioma stem cells and glioma endothelial cells is a key driver to tumor growth. We can therefore say that the perivascular niche can become a hub for convergence of multiple phenotypes, including self-renewal, maintenance, angiogenesis, and proliferation. Now, distinct phenotypes of glioma stem cells, we can see here, can either migrate across gradients of adhesion molecules and be tethered to the niche by haptotaxis, or exposed to chemotaxis gradients of soluble factors resulting to a prototype system of natural bio-nanomachines. And deeper insight into the perivascular niche reveals a prototype model of application layer and a series of bio-inspired test beds of functionalities for molecular communication applications. Support at the physical layer is achieved by glioma stem cells that serve as mobile nanomachines under the chemotactic control of detected concentrations of growth factors and migration along gradients of laminin, which is a natural polymer found in the basement membrane of the vessel. Structure for formation is an overarching functionality attributed to the spatial relationship of bio-nanomachines within these three-dimensional units, building a reaction diffusion model based on activators and inhibitors. Basic functionalities now can entail bio-nanomachine location and control, where, for example, interleukin-8, a known cytokine submolecules which regulate the mobility of cells, serves as attractor for glioma endothelial cells. Microenvironmental Q monitoring with emphasis on monitoring of molecular communication environment, and the tethering of glioma stem cells to the membrane via complexes of integrin and adhesion molecule with laminin. and ultimately control of molecule concentration, such as the delivery of growth factors. The strong message we can receive from the past three slides is that in order to find the most appropriate nanonetwork validation model, it is essential to recapitulate the in vivo microenvironment. In particular, to validate human molecular communication theoretical models, we do need to build basic testbeds enabling quantification of model parameters, but most importantly, need to first answer what do we want to measure, how we want to define, and how we want to analyze molecular signal metrics. So for successful disease diagnostics and therapeutics, we need detection and quantification of appropriate marker for disease. This is the sine qua non of oncology. We need deep knowledge of the spatial temporal and disease evolution. And we have to define, design, and synthesize suitable bio-nanomachines and nanonetworks. Coming to tumor pharmacology, which is a central discipline in therapeutics, we realize that it needs to be transformed to a new discipline where drug development testing models are directed by the concepts of molecular communication. In this context, therapeutically engineered cells as natural nanocarriers can be developed as prototype bio-nanomachines and ultimately recapitulate the in vivo conditions, applying the modes we have already discussed for molecular communication. Therefore, the challenge we are now facing for swift translation from the theoretical model to the clinic is how to translate the layered architectures to multi-layered tumor models. Three-dimensional tumor models comprise a significant technological advancement of the last decade and serve as valuable test beds in molecular communication because they combine tissue engineering, cancer biology into a molecular communication environment. Three-dimensional cancer models is the outcome of the implementation of a diversity of methods such as three-dimensional spheroids of cells, bioprinting, assembly of biomolecule, and biocompatible materials such as hydrogels, scaffolds, or even their combination into hybrid systems like this one of our own work hosting glioblastoma stem cells, and provide unprecedented backgrounds for the development of neurotaxis via physical factors, aptotaxis and topotaxis with microenvironmental parameters, and chemotaxis via gradient concentrations of different chemical factors. So, in an effort to take a big leap forward and take molecular communication to a drastically new level, 7 teams across Europe and Far East, led by the University of Cyprus and Cyprus-based nanobiotechnology SME, joined visions and forces towards the next generation theranostic of brain pathologies with autonomous, externally controllable nanonetworks. The Glegade or Fedel pen is the first international, interdisciplinary, far-sighted, integrated research approach in externally controllable molecular communications and has used the Circle HET-CSA as a strong networking foundation. It can be considered as a paradigm shifting interdisciplinary taking, introducing a working prototype of a complete, autonomous, and clinically applicable network-based molecular communication platform. An overarching science to technology breakthrough in Gladiator is the study of glioblastoma as integrated interactive networks of cell and exosomal bio-nanomachines in the tumor microenvironment. exosomes and extracellular vesicles are natural nanostructures of cell membrane encapsulating biomolecules such as RNA, DNA, protein, and they are released by a function known as exocytosis and uptaken by other cells via endocytosis. Exosomes can therefore build prototype nanonetworks, communicating information among cell bio-nanomachine, such as glioma stem cells, glioma endothelial cells, astrocytes, and tumor immune cells, such as the tumor-associated macrophages or T cells, maintaining integrated interactive networks in the tumor microenvironment. In Gladiator, exosomes as natural nanoparticles and bio-nanomachines can be subjects to external control of radio frequency, for example, serving as the building blocks of a diffusion channel model where neural stem cells engineered to produce reprogramming, which means therapeutic, changing the function of a gene. This is reprogramming. Exosomes serve as transmitters and can release and diffuse to the brain extracellular matrix, which is here the communication channel, other exosomes which are uptaken by glioma stem cells serving as receivers and maintain bio-nanomachine to bio-nanomachine interfaces. So what Gladiator proposes is literally brain tumor management as a fully autonomous, externally controllable molecular communication nanonetwork, where orthotopic tumor foci of glioma tumor cells and glioma stem cells, implanted organoids of therapeutically engineered neural stem cells, and implanted sensors of optically active cells serve as communication network blocks building three distinct channels, and where the optical signals generated at the sensor cells by tumor-released monitoring exosomes serve as the out messaging interface. An external device translates this message to radio frequency signal that regulates the genetic stimulation of growth molecules found in the reprogramming cells. This externally controllable molecular communication network and individual components are integrated into a clinically applicable working prototype of bio-nanomachine diagnostics and nanonetwork therapeutics. The validating in vivo model comprises a brain with orthotopically planted tumors of both glioma cells and glioma stem cells, subpopulations, and autologous organoid, A here, of reprogramming cells releasing the rationally designed exosomes, the therapeutic exosomes, the implantable hybrid sensors containing cell-based sensors and electronics with the cell-based sensors grown on electrospan nanofibers and detecting the reporting exosomes released by the tumor cells. An external transceiver patch based on the skull, where the radio frequency transmitter is located, and a central unit with long-range communication, wireless audio area network integration, localization algorithms, and analysis and intelligent feedback algorithms. The enabling patch and interfaces, which include the radiation frequency mediated reprogramming and external control, converge to an emerging supradiscipline we now call radiofrequency genetics. We had optogenetics, and now we built a new area, the radiofrequency genetics, and established brain molecule, not just brain molecule machine interfaces. This slide summarizes preliminary achievements of Gladiator and address gaps and challenges in the molecular communication field that we have already discussed. Remember the slide with the challenges we had to face. I think we have met some of them. Partners at the University of Ulu and Fraunhofer, in interaction with my team at EBOS, constructed the basic structural units, genetic units, for monitoring and therapeutic bio-nanomachines. And this is a representative figure of glioblastoma release exosomes, monitoring exosomes, interfacing with the neural stem cells. Osaka University, jointly with the EBOS team, they already presented growing bio-nanomachine, a glioblastoma evolution simulation model. Waterfront Institute of Technology, together with National University of Technology of Norway, developed and already published a controlled therapy simulation model based on the release rate of therapeutic exosomes. 3D tumor construct of glioblastoma cells grown on hybrid nanofiber and hydrogel scaffolds by our team at EBOS comprise the so-called biomimetic molecular communication and biomed. Significant advances have come in and out messaging interfaces, and they are already available by the University of Cyprus and Fraunhofer engineering teams, and include the design and preliminary prototype of implantable optoelectronics, miniaturization techniques for antenna design, which employ biometamaterials in order to reduce the size of the arrays to practical sizes for head placement, and for evaluation of energy absorption inside the body. The optimized design is evaluated using a realistic three-dimensional model, including all the parts of the human head. And this work is complemented by screening for new genes under radiofrequency control jointly with EBOS. So, are we at the dawn of an industrial revolution? I think so. These advancements seem to be bringing us at the real dawn of a new era, brave new world. Why? Because ongoing collaborative research is taking molecular communication to a drastically new level. An important allusion to the potential of molecular concepts is exemplified by their role as the main technology for implementing advanced medical solutions. Novel strategies are emerging, enabling integration of diagnosis and therapy across major specialties and subspecialties of clinical practice. We have shown that we have highly interdisciplinary research integration converging to solutions for realizing nanoscale communications. Bio-nanomachine diagnostics and nanonetwork therapeutics emerge as a superdiscipline, leading to a true paradigm shifting in oncology. Specific advancements across disciplines are addressing predefined challenges and gaps, because we have standardization of architecture and simulation framework. Great steps are already taken by the Circle CSA toolbooks, and further opportunities for coordinate actions can be provided by schemes such as the cost action. Structural tumor units are now recognized as working prototype models for higher level functionalities. The go and grow paradigm in tumor evolution and the attractive, interactive exosome uptake release loops epitomize what we define as collective bio-nanomachine behavior. What else? Three-dimensional bio-inspired organoids and tumor constructs define biosimilar molecular communication models, taking and exploiting useful lessons from biology. Engineer bio-nanomachine functions under in-messaging, out-messaging control of optical and radiofrequency interfaces are realizing the Internet of Bio-Nanothings and are bringing the brain-molecule-machine interfaces at a radically new level where computing systems do detect, do localize, and do control molecular activities bidirectionally. And importantly enough, all this can be achieved with minimum human intervention, paving the way to radically new brain-computer connectivity approaches. The challenge for us, the clinicians, remain to build the necessary innovation ecosystem by attracting the clinical community and to stimulate translation to clinical practice. We need to revisit the regulatory environment and, most importantly, reform our higher education and training towards the new generation of molecular communication leaders. My driving force as a physician has always been that no matter how innovative and disruptive our work may be, this innovation should not be for the sake of the technology, should not be for us, the healthcare professionals, but for people's life and their future. And the ultimate goal is to transform all our discoveries to recoveries. The appreciation and funding of the Gladiator project by the European Innovation Council is nothing else but a vote of confidence for the whole molecular communication community, for all of you. The challenge ahead is explicitly defined by those who have evaluated this work, by the reviewers of this project, who quoted that this interdisciplinary project will create significant added value for participants in society. Project results will provide the keys to a complete new regime of fighting deadly diseases. So Gladiator will pave the way to new treatments of cancer, as was the discovery of antibiotics past surgery. And I think this is now becoming our new promise. Certainly no man is an island, and great visions need joint efforts. I therefore want to thank my partners at the Gladiator project, and I'm sure you agree there can be no creation without recreation. My extended team at EPOS and University of Cyprus for contribution to ongoing and research projects that have nourished this work. But I feel obliged to specially mention three valued colleagues. Associate Professor Ioannis Krikidis at the University of Cyprus, who introduced me to this mostly exciting field of wireless and molecular communication that has radically transformed my research prospects. Professor Kostas Pitris, a long-standing collaborator who has been the engineering mind in most of my projects, and ultimately a wonderful leader in molecular communication, Professor Tadashi Nakano, for his kind invitation that has given me this unique opportunity to share with you today my fantastic voyage through bio-nanomachines and nanonetworks. Thank you all for your attention.
Hello everyone, my name is Huang Li from the School of Electronics and Information Engineering, Tongji University, Shanghai. I'm very happy to be able to participate in this online conference. My paper's title is Clock Synchronization of Diffusion-based Mobile Molecular Communication Systems in Nanonetworks. Next, I will introduce my paper from the following aspects. In the introduction section, I will introduce the concept of molecular communication, clock synchronization in nanonetworks, and the current research of clock synchronization in diffusion-based molecular communication systems. In the second part, I will mainly introduce the system model of mobile molecular communication system and the clock synchronization mechanism proposed in this paper. Next are the numerical results and conclusions. So, what is molecular communication? Molecular communication is an emerging communication method using molecules or particles as single carriers, which enables nanomachines to send messages at the nano or micro scale for information exchange and collaboration. In a nanonetwork, the capabilities of a single nanomachine are limited. Molecular communication makes nanomachines promising to expand the capabilities through information exchange and collaboration. Clock synchronization between nanomachines play an essential role in collaboration. For example, in a targeted drug delivery application, if the clocks of nanomachines are not synchronized, when multiple nanomachines release drugs to attack cancer cells, nanomachines cannot respond at the same time, and the drug treatment effect will be affected. But in current researches, it is always assumed that the transmitter and receiver in the MC system are synchronized. However, the clocks of nanomachines are not all synchronized automatically. The clocks of different nanomachines may be different. There may be a clock offset between different nanomachines. Some methods for achieving the synchronization of MC systems have been proposed. For example, the causal sensing mechanism of bacteria, inhibitory molecules, but these two methods need to correspond to specific molecules or cells. Besides, multiple rounds to achieve system clock synchronization is proposed, but many of them are based on fixed MC systems. At the same time, when exchanging clock information, the clock information is encoded into the molecule, but the synthesis time of molecule is not considered in the method. In addition, the movement of the transmitter and receiver in the MC system is an ubiquitous situation. The distance between the transmitter and receiver is constantly changing. The clock synchronization mechanism based on the fixed molecular communication system is difficult to directly apply to the mobile molecular communication system. So, effective clock synchronization mechanism for mobile MC systems in nanonetworks is necessary. In this paper, we consider an abundant three-dimensional fluid environment with constant temperature and viscosity. The Intel nanonetwork include a clock reference nanomachine and multiple nanomachines that require clock correction. In this paper, the clock reference nanomachine is modeled as a Separable transmitter. The nanomachines that require clock correction are modeled as receivers with multiple receiving antennas which are passive observers. Monoculars can enter and leave the passive observers freely. The transmitter nanomachine and the receiver nanomachine obey the Brownian motion. The transmitter can release multiple types of monoculars, and the receivers have multiple receiving antennas, and each antenna can detect multiple types of monoculars. As the figure shown, color bubbles represent different types of monoculars. The channel impulse for the number of released monoculars Q can be expressed as SIT tau. As we can see, the SIT tau is related to the equivalent diffusion coefficient, the distance between the transmitter and the receiver at the moment of monoculars release, and the propagation time of monoculars. We have considered the Brownian noise, which is subject to a Gaussian distribution with mean zero and variance SIT tau. Therefore, the number of monocular arriving as antenna is SIT tau plus noise. Assuming that there were M antennas, the number of monoculars received by the receiver is the average of the M antennas. When M is large enough, cause of noise follows a Gaussian distribution with an average value of 0. The average number of noise is 0. Therefore, the effect of Brownian noise on the system can be weakened. In a mobile molecular communication system, as the figure shown, we assume that when the system starts clock synchronization, the clock of the transmitter is TT0, and the clock of the receiver is TR0. So the clock offset between the transmitter and the receiver is phi. Just like the clock synchronization mechanism in other MMC systems, we also consider sending the clock information of the transmitter to the receiver. The receiver receives the clock information of the transmitter and adjusts its own clock so that the receiver and the transmitter are synchronized. But encoding the transmitter clock into monocular takes time. Therefore, as the figure shows, the release time of monocular is TT0 plus epsilon. In mobile molecular communication, since the transmitter and the receiver are constantly moving, the distance D between the transmitter and the receiver is unknown. The molecular propagation time is also unknown. Besides, because different clock value will result in different molecular structures, the diffusion coefficient of A moleculars is uncertain. At the same time, because the synthesis time of monocular is constant, epsilon is uncertain. Therefore, at the receiver, because of the synthesis time of epsilon of the monoculars and the clock offset phi between the transmitter and the receiver are uncertain. Even though the propagation time tau of the monocular is known, it is impossible to know the clock offset separately. Since the diffusion coefficient is uncertain and the synthesis time of A monocular is uncertain. In addition to the A monocular, another different I types of monoculars with diffusion coefficient Di are also used. They are released at the clock T0 of the transmitter. Therefore, for these I types of monoculars at the receiver, only the clock offset between the transmitter and the receiver, the propagation time of monoculars, and the distance between the transmitter and the receiver at the moment when the I-type monoculars are released are unknown. Since the number of the monoculars received by the receiver is subject to the arrival rule of the channel impulse response (SIT tau) in the mobile molecular communication system, the formula of propagation time can be substituted into the channel response formula. The receiver samples the number of monoculars that arrive. The number of type I monoculars arriving at the receiver at time Ti is sampled. Then the least square method can be used to fit the type I collected monocular number and the unknown distance between the transmitter and the receiver. For the type I plus 1 monoculars, estimated phi and distance can be used as the initial values of the least square method. For I types of monoculars, the final estimated clock offset and the distance is the average of I values. The clock synchronization mechanism is measured by the mean square error of estimated clock offset. The table on the right is the parameters used for simulation. As shown on the left figure, the relationship between the initial distance and the MSE of the estimated clock offset is shown. I represents the numbers of types of monoculars used. We can see that the performance of clock synchronization mechanism is affected by the distance between the transmitter and the receiver at the moment when the monoculars are released, and the types of the monoculars used. As the distance increases, the MSE of the estimated clock offset increases. This is because as the distance gradually increases, the number of single monoculars received by the receiver gradually decreases, and the received signal is gradually increases by the influence of noise. The more types of monoculars released, the better the estimation performance of the method. This is because the I types of monoculars are released at the same time, so the initial distance and clock offset between the transmitter and the receiver are the same. Different monoculars are affected by noise differently. Using multiple types of monoculars can reduce the effect of noise on overall synchronization performance. In addition to evaluating the clock offset between the transmitter and the receiver, we also evaluated the estimated distance. As with clock offset, the estimation of distance is also affected by the distance and the types of molecules used. We also compared the performance of clock synchronization methods in mobile molecular communication systems under several different scenarios. As shown in the figure, we compared the performance of the blind synchronization algorithm in the mobile molecular communication system proposed by other articles, compared with the blind synchronization algorithm. The proposed synchronization algorithm in this paper has better performance. This is because more types of molecules are used in this paper, and the clock of the transmitter is directly transmitted to the receiver. We also compared the performance of the clock synchronization algorithm proposed in this paper to two different scenarios. One scenario does not consider the synthesis time of molecules. It can be seen that the clock synchronization performance obtained after considering the effect of the clock synchronization It can be seen the clock synchronization performance obtained after considering the effect of molecular synthesis time is better. Therefore, it is important for the clock synchronization performance to consider the molecular synthesis time. However, the clock synchronization algorithm proposed in this paper applies more types of molecules. This relatively increases the performance requirement for transmitter and receiver nanomachines. In conclusion, we investigate the clock synchronization between the transmitter and receiver in a mobile MC system. The clock offset and the distance between the transmitter and receiver is estimated by releasing multiple types of molecules. The transmitter clock value is encoded into single molecules. The synthesis time of molecules is taken account. The clock offset between the transmitter and the receiver is estimated by the least square method. The initial system distance and the number of the types of molecules released will affect the performance of the MC system. In our future work, we will continue to study clock synchronization mechanisms and performance in more practical mobile MC systems in nanonetworks. That's all. Thank you.
Okay, let's get started. Greeting. This is a paper about the Cooperative Communication module of the Molecular Communication. For the abstract, we have mentioned the traditional drug delivery is knocked in delivery efficiency. Due to this, we assume there's another particle could be controlled in movement, and it could need the drug particles or other particles we interested to the target location. Then we could improve the module's efficiency by this method. In this module, we call the nanoparticle as the attractant. It could attract the other particles to enhance the concentration of the drug. This is our main idea about the whole module. Its properties about cooperative communication is that we control the attractant directly instead of the drug particles. So, we could control multi-particles to enhance the effects, just like the MIMO communication system. In my own view, and if this module is achieved, we could improve the efficiency of target drug delivery and using this module into particle medical tests. So this is an abstract. Let's proceed the introduction. The part of introduction, we have introduced some realistic problems in the drug delivery. And you can see the drug delivery is hardly improved these years because of its low efficiency. So this is our iteration to build this module. In this module, we consider the environment as a molecular communication system. You can see here, here is it. And the drugs could be considered as information of drug. You can say the next here it is. And so our goal is it shall be transported from the interjection site to the target location. Here it is. Before our building of the module, there are some researches about how to transport these drugs, including using the magnet-sensitive particles to lead the drug carriers and like the bacteria to the destination. You can say here is the latest research about this. Okay, let's proceed. So in this module, we assume the attractant as the source node and the released particle is the relay node in the cooperative communication system. You can say here is assuming, and in the about the application, we can divide it into two aspects. One is the drug delivery. We can use this to deliver the drug precisely, and the two is to clear the bacteria in damaged tissue. In this, we could consider the attraction and lead the bacteria to exit the human body. Okay, so this is its application. Let's proceed. Let's go to look at the picture. You can see there are many factors could disturb or have negative effect to the attractant, such as the bacteria, the barrier, and some factors in the vessel. You can here is factors. These matters could cause more complex problems and more compute. And so in this module, we would ignore these problems and assume the ideal and static environment in this paper and counter these problems in the future vision. Okay, let's proceed. The next part is the concept of how our module under the equation about our module. First of part is some concepts about the molecular communication, and so let's skip it and to the next part. The next part is the equation we used in this module. The movement of the attractant we consider it can be controlled by the magnetic and the electric field or a source. The attractant is small enough, and so we can equal itself as a mass point with the electron. In this module, the electric field would control the velocity of the attractant, and the magnetic field could control the direction of the attractant. And at last, we have set the sense upper limit to each bacteria. Once the concentration of the attractant particle beyond the sensing limit, we could consider the bacteria is attracted. You can say this is just the equations and how the position updates and works. Okay, let's proceed. And so in one word, the more bacteria could the module be attracted, and the less time have been costed, the module is more effective. All this function is based is a basic electromagnetic equations, because we consider all the particles as the mass point. So in future vision, we would consider the factors could affect the movement. So the section 3 is the result we have tested. The first one is we have tested a static environment. You can see this picture. We put the attractant in the static environment. You can see here is the attractant in the central position, and there shall no velocity and no other factors in the vessel, just bacteria's movement in the environment. Assuming there are bacteria randomly generated in the two areas, you can see here, the bacteria randomly generate, and the attractant stay related static in the environment. The goal is the bacteria find the attractant as quick as it possible, or the attractant could attract the bacteria as quick as it possible too. It's certain that the efficiency is very low and not stable enough. You can see the result. Here is the result of the first module. You can see the empty part means the bacteria didn't find the attractant. You can see it's pretty not stable enough, and you can see it cost different time. So you can see the randomly action can hardly locate and move to the destination. Due to this, we design another module. You can see here. The equation 8 is the gradient search. We consider the attractant could move by the effect of the magnetic and the electric field. They could cause the particle with a simple movement, and one of the bacteria would follow the equation 8, and another one could not. It did the randomly action. So based on this, let's see the result. Here is the module, and here is the result. The left one you can say is highly stable, but it cost much time than right one. It didn't find the attractant for only once, but you can see the right one didn't find for the attractant for several times. However, you can say that the group right is improved than the upper one. You can see it's more stable. So this is the two modules. You can see there are some problems exist in this module. The module is totally static and ideal. It doesn't consider the spread of the attractant and the exit of the bacteria, so it's not precise. And the real condition, the environment and the bacteria will not follow the gradient search, so we have to improve the algorithm. So in our future vision, my leader consider that we can improve and reforge this module again in the future vision. So this is our work in this paper. The next is just some conclusion and the result of our module. So you can see this is our conclusion. Our future version is going to use the DDPJ and put in a GUI for the module. So I think there's much thing to improve this module. So thank you for your listening and your attempting. Thank you.
Hello everyone, my name is Lu Zhiqiang and I will introduce my paper to you. Performance of diffusion-based MIMO molecular communications and dual threshold algorithm. I will use the following 5 parts to introduce: background, system model, ISI and ILI, receptor analysis, and simulation. Firstly, background. Molecular communication and multi-input, multi-output technology are the two nuclear research directions in this paper. In recent years, there is more and more attention being paid to molecular communication, which is defined as the molecules are used to physically carry the information. In MC systems, nanomachines are considered as the most fundamental functional unities that are able to perform simple tasks such as computation, sensing, or actuation. Furthermore, it proposed that the idea of forming a nanonetwork by interconnecting several nanomachines. As for MC, information can be encoded onto the molecules in different ways, such as CSK, MOSK, MCSK. As for multi-input, multi-output technology, we also call it MIMO. It obtains the gain of reliability, and validity is obtained mainly by using space resource, at the cost of increasing the processing complexity of the sending end and the receiving end. If we want to use it in MC, we need to solve the main interference, which comes from intersample interference and interlink interference. Now I begin to show the paper's main body. System modern part. The considered MIMO molecular communication system is composed of two transmitters, TX1 and TX2, and a receiver with two receptors, RX1 and RX2. TX1 is related to RX1 and unrelated to RX2, which means there are two interactional links. Each receptor is assumed to be spherical with radius. The distance of related transmitter and receptor is defined as d1, and the distance of unrelated transmitter and receptor is defined as d2. Also, each transmitter is assumed to perfectly control the emission process of the molecule into the innovation environment. CSK is the coding scheme of this system, as the two transmitters TX1 and TX2 are considered to release N1 and N2 molecules for transmitting the bit 1, while transmitting the bit 0 means that no molecules will be released in current slot. And in this system, transmitters are interrelated, which means transmit the same binary sequence simultaneously. As for diffusion-based channel, due to the random motion of the released molecules in fluid medium, the time that the molecule arrives at the receiver is probabilistic. We can get the probability that a molecule is absorbed by the receiver within time slot duration by Fick law. In this paper, we assume that there is no interaction between the molecules. So we can use binomial distribution to get the number of molecules which are absorbed by the receiver within the time slot and its approximate expression. ISI and ILI part. Some of the released molecules may arrive at the receiver after their intended time slot, which can lead to the information molecules of the subsequent transmission intervals are more than expectation. And we call this phenomenon ISI. In this paper, we consider that there is no interaction between time slots and its equal probability of transmitting bits 1 and 0 in any of the previous slots. Considering the complexity and accuracy of the formula, we can get a reasonable approximation for the ISI by only relating interference with several time slots. Interlink interference is caused by that the molecules released from the transmitter propagate randomly in the fluid medium based on diffusion, which causes that those molecules may reach at this related receptor and are absorbed by it. Compared with ISI, ILI needs to consider the molecules which are transmitted by the other transmitter in current time slot. So we need to discuss between transmitting 0 and 1. Receptor analysis part. The molecule received by Rx1 are considered firstly. It consists of two parts, from Tx1 and from Tx2. If transmitter send bit 1, the parts from Tx1 are ISI from Tx1 and probability distribution of which Tx1 send bit 1. The part from Tx2 is ILI from Tx2. If transmitter send bit 0, the part from Tx1 is ISI from Tx1. The part from Tx2 is ILI from Tx2. Then, bit L rate is considered. We take several steps to choose the threshold of single reception judgment. First, we set reasonable hypotheses on number of molecules received and apply likelihood ratio test. Then the expression is set to 1 to get the threshold. if the received number greater than the threshold, we believe the hypothesis that the transmitter sent bit 1. If not, we believe the other hypothesis. So the BR comes from the probability of N0 greater than the threshold and N1 less than the threshold. In the case of two receptors different result, dual threshold algorithm is proposed to choose the better result and reduce BR. As for the decisions of two receptors, we have enough confidence in their judgments. If both of the decisions are same, and choose it as final decision. However, if both of decisions are different like 0-1 or 1-0, the handling method is proposed to use a new threshold for the receptor where decision is 1, and trust the new decision as final decision. New threshold and single threshold have directly proportional relationship. The proportionality coefficient can be set by two ways. One is constant coefficient by simulation compression, and the other is adjusted dynamically. In this way, we choose other decisions parameter to adjust it. Simulation part. In this part, we present the simulation results for the error probability of the proposed MIMO transmission schemes over the diffusion channel. We consider the short-range molecular communication, where communication distances are typically within tens of micrometers. Throughout the simulations, we set default parameters, and you can see this in the paper. The first picture is the BR performance of different schemes versus the number of released molecules, which represent the transmission's power under the numerical results obtained from the number of correctly detected bits over the total number of transmitted bits, and the theoretical results obtained using derivation, and this shows the exactness of the theoretic analysis. There are 4 pictures on screen. Any of these show how one of the model-related parameter changes BR. On this picture, we can see that dual threshold decision performance better than single reception decision about BR very clearly. In the last picture, we plot the BR performance versus the number of released molecules, which represent the transmission power under many kinds of ways. Firstly, we observed that self-adapting dual threshold decision plays a better improvement function in reducing BER than every easy dual threshold decision. Then one of easy dual threshold decision is very close to self-adapting way, and we think that the associated coefficient is appropriate in easy dual threshold decision. In this paper, we studied the performance of a MIMO molecular communication system in a diffusion-based environment, proposed two dual threshold algorithms to reduce BER, and finally we used simulation to verify these results. That's all. Thank you for your watching.
Hello everyone, my name is Yutaka Okaye from Osaka University, Japan. Today, I'd like to talk about binary concentration shift keying with multiple measurements of molecular concentration in mobile molecular communication. Background: molecular communication has emerged as an enabling technology for nanoscale communication. Molecular communication is inspired by the communication among biological entities such as cells and bacteria. In molecular communication, molecules are used for information carriers. In encoding, transmitter encodes the information it wants to transmit onto the molecules. In sending, the transmitter releases the molecules into the environment. In propagation, the molecules released by the transmitter propagate in the environment. In receiving, the receiver receives the molecules. And finally, chemical reaction is induced in decoding. In today's talk, we focus on the modulation technique called binary concentration shift keying, where binary information is encoded on the molecule concentration. In this technique, transmitter denoted as Tx transmits either 0 or 1, where corresponding number of molecules are released. In many cases, more number of molecules are released for symbol 1 than that for symbol 0. Rx determines what symbol was transmitted based on the molecular concentration. When the concentration is greater than some threshold, Rx determines 1, otherwise 0. For example, this figure shows the molecular concentration that a receiver, denoted as Rx, receives as a function of time. The blue and red lines correspond with the symbol 1 and 0 respectively. Rx measures the molecular concentration at a pre-specified time, t, compare it with the predefined threshold, and then decides the information it received as 1 or 0. First, we introduce the mathematical model for the communication systems. The first model is about the molecular concentration Rx receives, where the mobility and counting noise are taken into account. The second model is about how RX demodulates the information from the molecular concentration RX receives. We consider two techniques, single measurement, which is existing work, and multiple measurements, which is what we propose in this paper. Here, we consider the molecular concentration Rx receives, mobility of Rx. The mobility here is a vibration or fluctuation in the location of Rx due to the thermal noise in the environment. In this case, the molecular concentration Rx receives is represented by the following equation. This equation is a well-known diffusion equation of molecules, where the volume of Rx is V, the number of molecules Tx released is A, the distance between Tx and Rx is R0, and T is the time passed since the molecules were released. dE is called the effective diffusion coefficient, given as the sum of the diffusion coefficients of molecules and Rx. By this equation, mobile molecular communication system is transformed into static one. For the molecular concentration that Rx measures is also subject to the noise, called the counting noise, which is expressed by additive Gaussian noise model in a couple of literatures. This equation says that the molecular concentration Rx measures follows the normal distribution with mean xt and variance sin xt, where xt is the molecular concentration Rx receives without the counting noise. Here I explain how Rx demodulates the information from the molecular concentration Rx measures. Before going into each of the techniques, let me explain the problem we consider here. Molecular concentration Rx measures as a function of time is shown in this figure. From the equation I explained earlier, this plot shows the concentration when Tx transmits 1, where the number of molecules Tx releases is represented by A1. The blue solid line is the concentration without noise, and the blue shadowed area is the counting noise for each time. In the same manner, the concentration when Tx transmits 0 is shown by red colors. Under these conditions, we would like to tell what symbol Tx transmits, either 1 or 0. For this problem, in the single measurement, Rx measures the concentration at pre-specified time, T peak, which is the time when the concentration peaks without noise. The molecular concentration distribution is shown in the figure on the right. The red one corresponds to symbol 0 and the blue 1, and the distribution functions are f0 and f1 respectively. In this case, the threshold concentration that minimizes the error probability is described by the equation above. The threshold concentration goes through the intersection point of the two distribution functions, f0 and f1. We developed multiple measurements for the problem. In this technique, Rx measures molecular concentrations multiple times at the time interval tau, then find the maximum concentration and its distribution functions for symbol 0 and 1, which are G0 and G1 respectively. For the determination of threshold is the same as the single measurement. We conduct numerical experiments to evaluate the performance of multiple measurements in comparison with a single measurement. We use the default parameters values in the table that we find in literature. These values assume endothelial cells for Rx and vascular endothelial growth factor VEGF for information molecules. For performance metrics, we use achievable transmission rate described by this equation. This figure shows the impact of effective diffusion coefficient with x-axis log scale and y-axis achievable rate. Solid lines show the result of multiple measurements, and dashed line shows the one of single measurement. In the multiple measurements, the measurement time interval tau is varied. The figure shows that the achievable rate stays unchanged regardless of dE in the single measurement, whereas it decreases in the multiple measurement. This is because the peak concentration doesn't change even if dE increases, which explains the result of single measurement. On the other hand, the time for molecular concentration peak becomes shorter as dE increases, which makes it difficult for multiple measurements to detect the higher concentration, resulting in the degrade in performance. We see that when tau is large, say 0.1 and 1, the performance of the multiple measurement becomes worse than the single measurement when dE becomes larger than some values. This figure shows the impact of the number of molecules when Tx transmits 0, which is represented by E0. The number of molecules are fixed to 100,000 when Tx transmits 1. As A0 increases, the performance degrades. This result is expected because the molecular concentration Rx measures for symbol 1 and 0 become the same with higher probability as E0 increases. This figure shows the impact of a measurement time interval, tau, with x-axis log scale. The result of a single measurement becomes constant since tau is not a parameter of a single measurement. The performance of multiple measurement degrades as tau increases because it is less probable that multiple measurements detect the higher molecular concentration. We find that multiple measurements outperform the single measurement when tau is less than 0.4 hours. Practically speaking, this time length is large enough for the receiver cells to measure the molecular concentration when we assume the receiver is a perfectly monitoring sphere. Let me conclude. We developed binary concentration shift keying with multiple measurements of molecular concentration. Then, compared with a single measurement in terms of achievable rate, and identified the advantageous conditions for multiple measurements with realistic parameters. Future work is to extend the model to multiple symbol transmission. In that case, we will have the issues related to mobility of transmitter and intersymbol interference. That's it. Thank you very much for your attention.
Hello everyone, my name is Ye Xiangyun. I'm a student from School of Electronical Engineering and Automation, Shanghai University. And today, the theme I'm going to share with you is causal network analysis and fault root point detection based on symbolic transfer entropy. The content can be divided into four main parts. The first part is introduction, the second part is method, the third part is case study, and the last part is conclusion. The first part, introduction. Fault diagnosis is the detection and root cause identification of abnormal events, which is a complex and time-consuming task. Recent genes emphasize improvements in the speed and accuracy of fault diagnosis as the most immediate potential benefit of industrial process monitoring. It is not only related with the safety of workers, but also with the economy and operation of the plant. Causal analysis techniques can be used for data-based fault diagnosis. The symptoms of a fault propagate throughout the process along these causal relationships. Therefore, the inferred causality can indicate the path of the fault throughout the process. While researchers have studied many changes in causality analysis in fault diagnosis, including transfer entropy, which is the main theme today, granular causality, cross-correlation, partially directed coherence, and convergence cross-mapping. Generally speaking, the mutual transmission of information in industrial information systems will change the complexity of variables, and this chain tree can help us analyze and study the causality between variables. The cloud in Channel is the person who first proposed the concept of information entropy in order to quantify the complexity between variables. And Stramberg is who further proposed the concept of transfer entropy, which unifies the complexity of the signal and the transfer of information, and quantifies the exchange of information between systems. Bao applied the method of transfer entropy to the study of disturbance propagation path maps of chemical process to find the root cause of the disturbance, and proposed a method to optimize the transfer entropy parameters based on the historical data in the field of process industry. After all this, we can know that TE is a model-free method based on data-driven information theory. It can obtain causal relationships between variables. It has been used for modeling, monitoring, and fault diagnosis of complex industrial processes. It can detect the causal relationship between variables without the need to assume any underlying model, but its calculation process is complicated and the calculation time is very long. Therefore, many researchers have built up methods to solve this problem. For example, Nanonetwork, he proposed the use of symbolic processing to reduce the number of computation operations in iterative-based simulation methods and accelerate their computation. The validity of the algorithm is varied by two examples, and the estimates of second-order statistics in wireless channels. This method can be easily extrapolated to another application that requires calculations in a single simulation run. The symbol transfer entropy is robust and fast to calculate, It can also quantify the dominant direction of the information flow between time series with identical and non-identical coupling systems, thereby improving the accuracy of causal paths. Through the symbolic transfer of entropy, a causal network diagram can be obtained, and the root cause of the fault can be found. After all the concepts mentioned above, we can go into the second part, the method part. At first, we can take a look at the transfer entropy. According to the information theory, the formula for the transfer entropy from x to y is like below, TE x to y. In this TE method, a canal function method or histogram can be used to estimate the probability density function, which is a non-parametric method that can be used to fit a distribution of energy. For discrete time series, however, the discrete transfer entropy of x to y is like the expression below, and similarly, discrete transfer entropy of y to x is like the expression below, TE y to x. Then we can go into the symbolic transfer entropy. The symbol is defined by reordering the amplitude values of the time series xi and yi for a given time series xi. So the time series in ascending order of the amplified, like this expression. If the amplitudes are equal, then we can obtain this equation, and we can know that KIE is smaller than KI2 can be written as the following expression. This expression is to ensure that each xi uniquely maps to one of the possible arrangements of the factorial of the m. Therefore, the sequence can be presented by a symbol xi caret. Given a symbol sequence xi caret and yi caret, the symbol transfer entropy, which is STE, can be defined as the following two expressions. Then we can go into the third part, which is also the last part, significance test. The value of the transfer entropy represents the causal relationship between the variables, and if x is the cause variable of y, then the transfer entropy value between x and y must be greater than 0, otherwise it must be 0. In the actual situation, the system is also affected by various interference and noises, so that the value of the transfer entropy will not be exactly equal to 0. Therefore, a threshold needs to be set as a standard to determine whether the obtained transfer entropy is significant. Transfer entropy result above the threshold value is considered to be significant, and the transfer entropy result below the threshold value is considered to be not significant. In this paper, we choose the method of Dwan to calculate the significant threshold. To scramble the sequence of xy at the same time, and then construct a new sequence as follows: xs and ys. And the range of values of I and j is from 1 to n minus n plus 1. Only in this way can the original sequence equal to the new sequence. At the same time, in order to ensure that there is no correlation between the original sequence and the new sequence, I and j should satisfy this equation. In this equation, E is a sufficiently larger integer, and its value should be much larger than the prediction range h of the transfer entropy. It can be considered that the correlation between the two variables is eliminated by the long-time interval between the variables. Thus, two variable sequence without causality of 10, so as other groups, and the transfer entropy value of the sequence is calculated as TES, which is put into NTE, and here is the NTE's expression. And the significance threshold is calculated as follows: Sy2x is equal to mu NTE, which is the mean of the NTE, plus 3 double theta NTE, and the theta NTE is the variance of the NTE. Then we can do a simulation to test if our concepts are right. Well, we take the tendency estimate process as an example to conduct an experimental study to illustrate the specific modeling process of the causal network modeling method based on symbolic transfer entropy, and to verify the effectiveness of the proposed method in fault diagnosis. First, let me introduce you the background of the case. The duration of each TE process simulation in this case is 48 hours. And second, the fault is introduced into the simulation after the 8th hour. And third, each simulation will generate 960 observation samples. Among them, 160 samples are normal and 800 samples are faulty. In the simulation, the fault 7 in the TE process is due to the pressure loss in C in string 4. These conditions are what we all know about the case. And then we can do the simulation. Firstly, the variables that need to be subjected to the transfer entropy causality analysis are selected based on the contribution map of the PCA. The table below is the transfer entropy results for fault 7. And in each cell in the table, there are two rows. The first row of the data represents the transfer entropy value, and the second row of data represents the significance of Monte Carlo. Secondly, according to the map of the PCA we obtained above, we can draw path propagation diagram. Here is the path propagation diagram of fault 7. It can be clearly seen from the diagram that the root cause of the fault 7x is x4. This is in line with the actual situation, and the above illustrates the effectiveness and efficiency of the symbolic entropy. Therefore, our concepts and the simulation, they are right. Now, at last, we can conclude the main points of this paper. First, a model field approach is proposed to transfer entropy based on data-driven information theory, capable of calculating causal relationships between variables, and has been used in the field of modeling, monitoring, and fault diagnosis of complex industry process. Second point is, The symbolized signal is used to find the value of the transfer entropy between two variables. When the transfer entropy value between the variables is greater than the threshold calculated by the Monte Carlo significance test method, it indicates that there is a causal relationship between the two variables, so that the causal relationship can be obtained. The third point is, through the simulation and actual industrial case TE, Tenancy Eastman process, the correct root cause of the fault is found from the quantity diagram. And that's all. Thank you for listening.
Hello everyone, my name is Chen Zeng. I come from the School of Mechanical Engineering and Automation of Shanghai University. The topic of my presentation is personalized EEG feature extraction method based on filter bank and the elastic network. The main content is divided into the following 4 parts. The first part is a brief introduction. The second part is an introduction to some commonly used algorithms. The third part is the implement of method proposed in this paper and the analysis of the experimental results. The last part is the conclusion. The first part is an introduction. First of all, I will introduce 2 concepts, EEG and BCI. As we all know, the brain is a system with complex structure and function. It consists of hundreds of millions of neurons, and each neuron relies on the form of electrical signals to transmit information. We call this electrical signal as EEG signal. BCI. The brain-computer interface is a new human-computer interaction method based on EEG signals. It can transmit directly the human brain signals by not transmitting through the channels composed of peripheral nerves, muscles, and brain, and relies the interaction and control of the human brain and external devices. So a typical brain-computer interface system should be able to quickly and accurately extract and identify EEG information, reflecting different mental states of the human brain. This requires design a corresponding EEG signal processing method according to the specific situation. The EEG collected by the signal processing algorithm is analyzed and identified to extract the information reflecting the brain's thinking state, which can be divided into 3 steps: pre-processing, feature extraction, and classification. Pre-processing is to weaken the noise and artifact interference in the signal and improve the signal-to-noise ratio. The main methods are filtering, channel and frequency band selection, and so on. After pre-processing, the cleaner signal is beneficial to the subsequent signal processing. Feature extraction is to extract the main information that can reflect the intention of subjects from the pre-processed data. The classifier classifies the obtained characteristic information according to a certain criterion, and then converts the classification results into corresponding control signals to realize the control of external devices. Next, I will introduce several commonly used feature extraction and classification algorithms. Feature extraction algorithm. Feature extraction is the core of signal processing in BCR system. The quality of extracted features is directly related to the classification effect of subsequent classifiers and the efficiency of the whole BCR system. Due to the large amount of original signal data, the signal characteristics are not prominent enough. So it is difficult to get good classification results by directly use the original signals for classification. The purpose of feature extraction is to extract usable information from the EEG obtained in the preprocessing link, which can represent the corresponding conscious task, so as to obtain better classification performance. The following are three common methods for EEG feature extraction in BCR system of motion image. Common space mode method, wavelet packet decomposition method, and the power spectrum estimation method. Classification algorithm. Classifying the extracted EEG features is the final step of the BCR system and a very critical step. It maps these EEG signals reflecting the current mode of human activity to the specified classification and converts them into some control commands to control the external devices. The performance of the classification algorithm directly determines the performance of the entire BCI system. Generally, the performance of the classification algorithm is evaluated from the aspect of classification accuracy, operation rate, simplicity of the model, and interpretability. The following are three classification methods that are often used in EEG signal recognition: support vector machine, k-nearest neighbor, and the linear discriminant analysis algorithm. How to quick and effectively extract EEG features and improve recognition accuracy is the key to BCR technology research. Based on the comparative study of several common feature extraction methods, it is found that CSP feature extraction method has achieved good results in the brain-computer interface research based on motion imagination. However, the CSP algorithm itself lacks frequency domain information, limits the universality and the practicability of the CSP method. To solve this problem, CAP proposed a feature extraction method of filter bank common special pattern, FBCSP, which used the filter bank to divide the original signal into frequency bands and then use CSP to extract features in subband signals, thus enriching the frequency domain information of CSP feature extraction. Elastic network is a multivariate model analysis of method, which is the weighted balance of regression penalty and the lasso penalty. So it combines the advantages of the lasso and the ridge regression methods. By optimizing the model parameters, it is possible to find a balance between the goodness of fit and the complexity of the model to select an optimal model, which can make the fitted model more concise and improve the recognition accuracy. The third part is algorithm implementation and analysis. In this paper, based on the idea of frequency band segmentation, a personalized feature extraction method based on filter bank and the elastic network is proposed. This method has the following three steps. Firstly, get a high vector collection through band segmentation. The EEG is firstly divided into frequency bands by filter banks, and The EEG signals of each subband are extracted by a CSP special filter to obtain a high-dimensional feature set. Then, the elastic network method is adopted for feature selection. That is, when training the logistic regression model of elastic network with training data, the classification accuracy of classifier is taken as the evaluation standard. And the model parameters are optimized through tenfold cross-validation to obtain the feature subset with the highest EEG signal recognition degree applicable to different individuals. Finally, the test data feature set corresponding to the selected optimal feature subset is classified using the trained elastic network logistic regression model, and the corresponding classification accuracy is obtained. The experimental results shows that the proposed method is effective. The specific implementation of algorithm is mainly divided into two parts. One is the training sample and the other is the test sample. The first step is the training process. The training process have two steps: filter bank band segmentation and feature selection. In the first step, firstly segment into 17 subband signals using a Chebyshev type 2 band pass filter bank. Then input into the CSP special filter. Finally, extract the two-dimensional feature vectors to obtain a 34-dimensional feature vector set. In the feature selection steps, firstly, the elastic network logistic regression model is introduced to compress the obtained multidimensional feature variables. Then the feature subset with the highest recognition is selected for the individual's EEG signals. Specific implementation of test process. Firstly, as in the training process, the original signal is subband segmented using a filter bank, and the feature extraction is performed on each subband signal by CSP, so that a 34-dimensional feature variable set can be obtained. Feature selection. The test data corresponding to the selected optimal feature subset in the above training process is sent to the trained elastic network logistic regression classifier to perform prediction classification, and the classification accuracy of the set sample is obtained. Next, I will introduce experimental data source. The dataset I is derived from the data set 1 provided by the fourth brain-computer interface computation data in 2008. The dataset contains the EEG data obtained by the 7 health subjects for the motion imaging task. During the experiment, each subject performed 200 motion imaging experiments, and the effective time for each motion image was 4 seconds. The experimental process of motion imaging is shown in Figure 1. During the experiment, 59 channels of electrode caps were used to record EEG. The distribution of 59 electrodes in the brain region is shown in Figure 2. The sampling frequency is 1000 Hz. The data are only filtered by 0.05 to 200 Hz. Data is processed according to the algorithm mentioned above. Then, in order to prove the superiority of the encapsulated elastic network feature selection method combined with the filter bank proposed in this paper, it is compared with the conventional frequency band selection method and the filtered elastic network feature selection method. Based on the data of 4 subjects, the recognition accuracy of the 5 combinations obtained by the above several feature extraction and classification method were compared. In the right figure, as we can see, the method proposed in this paper have a highest classification accuracy. The last part is a brief conclusion. This paper analyzes and processes the motor imagery EEG data in the brain-computer interface system. The main contents are divided into the following aspects. Firstly, based on several commonly used feature extraction and classification algorithms in the current BCI system, the analysis and research are carried out. The best combination method to obtain higher calculation rate and recognition accuracy provides some theoretical reference for the practical application of BCI system. Then some shortcomings of CSP algorithms are improved. A personalized feature extraction method based on filter group and elastic network is proposed, which can eliminate the individual differences of EEG signals, realize automation, feature selection, and improve classification accuracy. That's all. Thanks for your listening.
Good afternoon, ladies and gentlemen. I'm glad to be here to make a presentation in this year's BICT meeting. My name is Qingying Zhao. I come from Changshu Institute of Technology. Title of my paper is Release Rate Optimization based on MMCC-Q in Local Nanomachine-based Targeted Drug Delivery. It's mainly for applications of molecular communication in targeted drug delivery. For targeted drug delivery systems, pegylation, passive targeting, and EPI effect, and active targeting are the 3 key technologies stimulated the development of nanocarriers in targeted drug delivery. The figure 1 shows these key technologies for targeted delivery of nanoparticles to the tumor. And today, macro and nano swimmers are proposed to autonomously detect diseases and deliver drugs with greater precision. The autonomous swimmers can be divided into biological systems, for example bacterial, sperm, and synthetic, for example nanorobot or nanomachine. After entering into the diseased site, nanomachines would release drug molecules to treat diseased cells. Depending on interactions between ligands on the surface of drug and receptors of diseased cells. Figure 2 shows the simplified separable shapes for a diseased cell and its receptor on the surface. There has been a drug response theory named occupancy theory associated with the ligand-receptor interactions. It is considered that the drug response is proportional to the proportion of receptors occupied by drug molecules in occupancy theory. However, the full drug response does not always require all receptors on diseased cells are occupied by drug molecules. Some types of drugs require only less than 10% occupancy of receptors and a least effective drug concentration to produce a full drug response. Accordingly, it could be an approach to estimate the optimized drug release rate of nanomachine according to the least occupancy proportion of receptors in order to decrease the loss of a drug molecule while maintaining a full drug response. Now, based on Ficker's law of diffusion, we can obtain the steady state molecular concentration CRT at a location distant from the emission point is R. In order to simplify the complexity of drug reception process, it is assumed that the drug reception process takes place inside a receptor space which has a spherical shape of a radius zone. The N receptors homogeneously distributed inside the receptor space in which the molecular concentration CRT is considered homogeneous at the same time t and equal to the value at the diseased cell location. The receptors can be changed between bound referred to as occupied by drug molecules whose bonding rate is key, and unbound referred to as unoccupied or undergoing bound internalization with the rate mu. Due to the continuous emission of drug molecules, it is considered that the molecular concentration CRT is not affected by the ligand receptor bonding. And at last, we assumed that the arrival process of molecules at the receiver is a boson process. We established the reception model of drug molecules based on ligand receptor and based on the previous assumptions. The change of the receptor state in the receptor space can only consider the change in the number of linked receptor pairs and free receptors. Therefore, the change of the receptor state in the receptor space can be represented by the Corrin model of MMCC. Here, the parameter n denotes the state of linked receptor bond number is equal to n. Parameter named n is the formation rate of linked receptor bonds, and the parameter mu n is the assimilation rate of linked receptor bonds. In the MMCC Corrin applied in drug reception process, the NT is a birthday discrete valued continuous time microprocess. The state transition probability diagram is shown in the figure 3. Where n denotes the number of bound receptors. The parameters named n. And the parameters mu n depend on the currently occupied state of the system, namely state n. Let probability Pnt be the probability that the population of linked receptor bonds is of size n at time t. That is, the system has n linked receptor bonds at time t. The dynamics of this Bost's process is that the set of differential difference equations, where the parameter named n represents the arrival rate of customs in Corrin systems when it is in state n. But in our drug reception process, it is associated with the poor receptor bonding reaction rate equal to the constant K. depending on the concentration of drug molecule, the CRT, and the number of occupied receptors in receptor space. Consequently, the coefficient lambda n can be formulated as the parameter mu n can be formulated as where the t is the average value of bound internalization time. Now, combining these equations, we can obtain the probability Pn. From occupancy theory, we can obtain the average number of occupied receptors Ns and the ratio of occupied receptors F. We also can obtain the least effective concentration CRT to produce a full drug response. And finally, the optimized release rate is derived. In order to exemplify the validity of the proposed method to optimize drug release rate, we established our simulations. The table 1 is the analytical average number of occupancy receptors derived from the optimized release rate equation. The single figure showed the variation of the number of occupied receptors in our simulations. Obviously, the numerically simulated number of occupied receptors approaches the analytical ones as time increases. Thank you.
Hello, everyone. I come from Shanghai University. This paper name is Research on Course Control of Unmanned Surface Vehicle, USV, with the application of USV in port patrol, environment monitoring, etc. The research is more and more in-depth of USV at home and abroad. One of the key technologies of USV is course control, because it determines the tracking error. I will introduce the paper from the following aspects. 1. Fuzzy PID controller. 2. Fuzzy control principle. 3. Hardware platform of USV. 4. Simulation and experiment. 5. Conclusion. The USV system has the characteristics of large inertia, long time delay and underdrive. The traditional linear control method is not robust enough to achieve coarse control. In this paper, a coarse controller of USB has been proposed based upon the fuzzy control theory. The following works have been completed. 1. Build mode of USB. 2. Parameter identification by least square method. 3. Build hardware platform. 4. Simulation and marine experiment. 1. Fuzzy PID controller. 1.1. Mathematical model. Select the second-order response model of NoModo to describe the plane motion mathematical model of USB. The formula is shown below, where delta is rudder, r is coarse angular velocity, k is gain, t1, t2, t3 is time delay constant. Parameters are identified by least square method. In this paper, the goodness of fit is used to identify result. The measure of goodness of fit is a determinable coefficient r, as shown in Formula 2. The value range of r is 0, 1, where yi is the sampling value. Y bar is the output value of identification model. 1.2. Design of coarse controller. The coarse controller of USB is based on the theory of fuzzy control. The structure design of coarse controller is shown in Figure 1. The input of the controller is the coarse deviation and the coarse deviation change. Output is actual coarse, where phi ir is expected coarse, E is coarse deviation, EC is coarse deviation change, delta is rudder, r is coarse angular velocity, phi ie is actual coarse. 2. Fuzzy control principle. The input of fuzzy control is coarse deviation and coarse deviation change. According to the rule table, the control value is obtained by Mamdani reasoning and fuzzy settlement, as shown in Figure 2 below. The structure fuzzy control include domain transformation, physicification, Mamdani reasoning, ambiguity resolution, and its input is E, and EC, output is U, where E is coarse deviation, EC is coarse deviation change, U is control value. 2.1. Fuzziness. In order to change the input variable into a fuzzy set that can be identified by the controller, it is necessary to deal the input with fuzzy measure. In this paper, the fuzzy set is divided into 7 subsets: PB, PM, PS, Z, NS, NM, NB. The triangle membership function is used to deal the input of coarse deviation, the change of coarse deviation, and the output of fuzzy control with fuzzy measure, as shown in Figure 3 below, where E, EC, U is domain transformation of E, EC, U. 2.2. Fuzzy rule table. The fuzzy rule table is obtained through the experience of manual operation. The control rule table with the output of each parameter is shown in Table 1 below. 2.3. Mamdani fuzzy reasoning. Using the Mamdani reasoning method, any group of fuzzy output sets of coarse deviation and coarse deviation change can be attained. The result of Mamdani fuzzy reasoning ergodic output is shown below. 2.4. Defuzzification. The output of the fuzzy controller is the exact value, and the solution of fuzzy is the process of transforming the fuzzy set obtained by fuzzy reasoning into the exact value. In this paper, bisector is used to solve the fuzzy problem, as shown in Formula 3, wherein is the number of fuzzy sets after fuzzy reasoning, MUI is membership value corresponding to the ith bisector of fuzzy set area. U is the exact result of defuzzification. 3. Hardware platform of USB 3.1 parameters. The research object of this paper is John Guai-2 of Shanghai University. John Guai-2 inches belongs to the category of water jet propulsion. It adopts the design principles of standardization, modularization and systematization, and it is applied to marine surveying and mapping. The figure 5 shows the appearance and internal structure of John Guai-2. 3.2 Composition of hardware system. The equipment carried by the John Guai-2 include INS, GPS, microwave communication, maritime radar, laser radar, forward-looking sonar, etc. The equipment for remote monitoring is mainly remote monitoring stations, as shown in Figure 6 below. 3.3 Remote monitoring software. The main function of remote monitoring software is route planning and USB dynamic information display. It can be divided into chart management module, route planning module, video monitoring module, obstacle information display module and USB integrated information module, as shown in the Figure 7. 4. Simulation and experiment 4.1 Identification result. Identification result of model parameters of John Guai-2 in 3 different speeds, as is shown in Table 3 below. It is shown that the coefficient of determination is not good at 1 to 4 kN. 4.2 Simulation. The result is shown below in the simulation experiment. The input signal source is step signal. The initial course is set as 0 degree. The expected course is 60 degree. The sampling frequency is 0.1. The random interference signal is added at 60s. The duration is 3s, and the range is 0 degree, 5 degree. As is shown below, the course change curve and the corresponding actual rudder angle change curve under random interference. 4.3 Experiment. The marine experiment was carried out in North Shipyard, Qingdao. The experimental area on the chart and scene are shown in Figure 9 below. 4.4 Experiment result. As shown below, it can be seen from the experimental results that after the start of control, the actual course of USB can rotate rapidly to the expected course, and finally vibrate near the expected course. The oscillation is caused by uncertain wind, wave and current. These external forces will make the USB course deviate when they act on USB. Practice shows that USB can still keep straight line operation effectively when the course vibration is within a plus or minus 10 degree. Conclusion. The simulation experiment of the model with 3 parameters shows that the control effect is good in medium and high speed. Furthermore, the hardware platform is built, and the experiment is done in the sea. The experiment shows that the design of the course controller worked effectively. Thanks for your attention.
Hello, I'm Chen Qixin from Shanghai University. I'm happy to share this article with you. Design and experiment of a double loyal vertical axis wind turbine. I will introduce it in detail from the following 6 parts. Chapter 1, Introduction. Chapter 2, Blade element theory. Chapter 3, Numerical simulation of aerodynamic performance of wind turbine. Chapter 4, Design of vertical axis wind turbine. Chapter 5, Prototype experiment. Chapter 6, Conclusion. The purpose of this article is to design a vertical axis wind turbine for mobile robots. Vertical axis wind turbines have many advantages. Number 1. The gearbox, generator, and controller in the vertical axis wind turbine can be placed on the ground with a low center of gravity, convenient maintenance, long life, and stable operation. Number 2. The vertical axis wind turbine is simplified structure because it can feel wind from any direction without a yaw device. Number 3. When the vertical axis wind turbine is running, the tip speed is smaller, cause less noise. Blade element theory is the basic theory of blade design. We find that the airfoil characteristics should meet 3 requirements through the blade element theory. Number 1. Large gradient of lift coefficient. Number 2. Small drag coefficient. Number 3. The airfoil can maintain excellent aerodynamic performance during the change of the angle of attack. The selection and design of airfoil is the key to the design of windmill. In this paper, a better airfoil is selected by comparing the aerodynamic performance of different airfoils. The airfoils NACA 0012, NACA 0021, NACA 4412, NACA 23012 are chosen. During the simulation experiment, the angle of attack is set to 0 degrees. The airfoil and wind speed are changing. The lift coefficient Cl, drag coefficient Cd, moment coefficient Cm, and lift-to-drag ratio Cl to Cd of 4 different airfoils are analyzed when the wind speed varies between 5 to 25 meters per second. As can be seen from the above figures, the lift-to-drag ratio of the NACA 4412 is the largest at the wind speed of 5 to 25 meters per second, especially around 70 meters per second. Its aerodynamic performance is better than other airfoils. NACA 4412 has a high lift coefficient and a small drag coefficient, and its excellent aerodynamic performance can be maintained in a large range of angles of attack. Therefore, the NACA 4412 is chosen as the blade airfoil of wind turbine. Other structural parameters of wind turbine are determined by the method of uniform optimization design. 12 groups of data are selected for experiments. and the results are shown in Table 1 below. We use MATLAB software to process the data obtained in Table 1. After calculation, when x1 equal 0.5, x2 equal 0.196, x3 equal 2.764, x4 equal 10. The stepwise regression equation obtains the maximum value. The radius of the wind turbine is 0.5 meters. The total length of the blade is 0.196 meters. The number of the blades is about 3. The mounting angle is 10 degrees, and the spherical wind energy utilization rate is 39.3%. After that, we use Inventor software to perform the 3D modeling of the wind turbine. The model mainly includes wind well, generator, and support. The oversized height of the wind turbine is 0.8 meters, and the radius is 0.5 meters. The wind well is designed in 2 layers, and the angle between the 2 layers is 60 degrees or 90 degrees respectively. So the wind from any direction can make the wind well generate a large torque, reducing the start torque of the generator and the starting wind speed of the wind turbine. Then we use ANSYS to analyze the static strength of the mode, apply the corresponding load on the wind well, and simulate the stress and deformation. It can be seen from the simulation results that the maximum strength of the triangle structural hub is 4.4 MPa, and the max deformation is 0.15 millimeters when installed at 90 degrees. The maximum stress of the triangle structural hub is 3.7 MPa, and the maximum deformation is 0.14 millimeter when installed at 60 degrees. The maximum strength of the 2 installation methods is far less than the respective yard strengths, and neither exceeds the allowable strength, so it meets the strength requirements of the material, which cause no damage when the wind turbine is running. Then we made a prototype for the experiment. The experimental platform of vertical axis wind turbine system is mainly composed of wind turbine system and air blower. A data acquisition platform is also necessary in the experiment. The data acquisition platform mainly include ammeter, voltmeter, anemometer, and speed detection system. The ammeter and voltmeter are used to detect the electrical output of wind turbines. The anemometer can measure wind speed, and the speed detection system measures rotational speed of wind well. First, the blade installation angle experiment was carried out to observe the output power and convection efficiency. The experimental results are shown in Table 2. We can see that the optimal blade installation angle is 6 degrees. At this time, the output power is 46.5 watt. Conventional efficiency is 18.75%. Then, the angle experiment of the wind well was carried out, and 60 degrees or 90 degrees were selected to observe the output power and the conventional efficiency. The experimental results with the angle of the wind well is 90 degrees are shown in Table 3. We can see that the output power increases with the increase of the wind speed, and the convection efficiency increases first and then decreases with the increase of the wind speed. When the wind speed is 7 meters per second, the convection efficiency is up to 24.43%. The experimental results are shown in Table 3 when the wind well. In Table 4 when the wind well angle is 60 degrees. As can be seen from the tables above, performance of the wind turbine at 60 degrees installation angle is better than 90 degrees installation angle, and the maximum convection efficiency of wind energy is 24.66% at the wind speed of 7 meters per second. This paper mainly presented a vertical axis wind turbine. Firstly, the aerodynamic performance of the wind turbine structural parameters was numerically simulated using CFD software combined with the uniform design experimentation method. The wind turbine parameters were determined. The wind turbine radius was 0.5 meters. The blade chord length was 0.196 meters. The number of the blades was about 3, and the installation angle of the blade was 6 degrees. Then, the wind turbine structure model was established, and the static strength analysis was performed using ANSYS software. The maximum strength obtained by the simulation was much smaller than the allowable strength of the structure steel, and the maximum deformation related to the overall size of the wind turbine was negligible. Finally, a prototype was fabricated to verify its performance. The prototype experiment results showed that performance of the wind turbine at 60 degrees installation angle was much better than 90 degrees installation angle, and the maximum convection efficiency of wind energy was 24.66% at the wind speed of 7 meters per second. So thank you for watching.
Hello everyone, it is a pleasure to introduce you to our work, and I will briefly introduce what we have done. The title of the paper is Real-Time Obstacle Detection based on Monocular Vision for Unmanned Surface Vehicle. From the title, you can note that the keywords of the paper include obstacle detection, monocular vision, and unmanned surface vehicle. First, let's take a brief look at the unmanned surface vehicle. The unmanned surface vehicle is a kind of unmanned surface platform which can sail autonomously in the marine environment. It can complete various maritime tasks intelligently. Obstacle detection technology is a key part of the environmental perception of the unmanned surface vehicle and is a prerequisite for its autonomously obstacle avoidance and safe navigation. The purpose of our algorithm is to detect the obstacle in front of the driving process through the image obtained from the unmanned surface vehicle's sensor, monocular camera, and guide unmanned surface vehicle to conduct autonomous and safe navigation. As mentioned earlier, the unmanned surface vehicle uses a monocular camera to capture video or pictures to obtain information about the surrounding environment. This page shows some photos taken by the monocular camera and is part of the test set for final testing. By using our algorithm, the final result can get the coordinate of the obstacle, which is shown in the yellow box in the figure after visualization. The requirement for obstacle is that they must be partially or completely in seawater. In order to avoid obstacle in time, our algorithm must be able to process the video captured by the camera in real time. Fortunately, the processing speed of the algorithm exceeds 33 frames per second, meeting the real-time requirements. On the right is a flow chart of our algorithm. Let's see the flow chart. In general, it is divided into 3 parts. One is image preprocessing, which is shown as a part surrounded by green box in the flowchart, and the second is water boundary line detection, which is shown as a part surrounded by red box. The last part is the saliency detection, shown in the figure as the part surrounded by the blue box. Next, I will introduce this main part separately. In order to adapt the algorithm to different sensors, we first scale and filter the capture image, where the filtering operation is to reduce the isolate noise in the image. Each pixel value is expressed as a 5-dimension vector, and the value includes 3 color components and 2 position components. Simple linear iterative clustering algorithm is used to process the picture into a collection of superpixels, so that superpixels can be the object of the subsequent algorithm processing. In the final version of the algorithm, we set the number of superpixels to 1200. At the same time, it needs to be explained that a good result can be obtained if the number is above 500. This is much less than the number of pixels in the original picture, and this can significantly reduce the processing time of the algorithm. And this is one of the reasons why our algorithm can achieve real time. Next, water-boundary line detection. Now we have a collection of superpixels. We assume that the image is divided into 3 roughly parallel semantic regions. The top is the sky, the middle is haze or land, and the bottom is the seed. We use a Gaussian mixture model based on microrandom file to model the superpixel set. This is an important part of our algorithm. In the paper, we spent a lot of space to introduce the mathematical principle behind this. You can find the complete process in the paper. The general idea is to use the Gaussian mixture model to model the superpixel set, and then introduce the microrandom file into the model, and then solve the iteration formula through the expectation maximization algorithm, and use the iteration formula to iterate the parameters of the model. By iterating on the model, we can use it to calculate the class to which each superpixel belongs, so as to achieve roughly semantic segmentation of the image. After visualization, as shown in the mask picture. But the goal of this part of our operation is to get the water boundary line. To achieve this, we first merge the top and middle area, and then we select a sufficient number of key points at the junctions of the two areas, and use the random sample consensus algorithm to fit the water boundary line. This page is the result of our water boundary line detection. Quantitative analysis can be viewed in the experimental part of the paper. The pictures shown on this page is also the result of the example picture on the third page of PowerPoint through our algorithm. You can see the effect is still relatively good. Regardless of the situation above the seawater area in the capture image, the obtained water boundary line can better meet the needs of subsequent processing. Finally, the saliency detection. On this page, the entire figure is a flowchart of saliency detection. Here is the beginning. Here is the end. Each small picture is the visualized effect pictured. The letter in the figure represents a small step. The table in the upper left corner has a brief introduction to each step. First of all, we already have a superpixel set and a water boundary line. So we can extract the superpixel of the C class below the water boundary line. Through this operation, we can reduce the amount of data processed by the following algorithm, thereby speeding up the exclude speed. Then process with foreground estimation and background estimation. Step B is the foreground estimation. This step is indicated by a red arrow in the figure based on the color comparison information. We calculate the probability that each superpixel belongs to the foreground. Thus, complete the estimation of the foreground. And step C to F are background estimation. This part is indicated by the green arrow in the figure. Step C is to extract the superpixel set connecting to the image boundary. Step D is to remove the superpixel set that may be obstacle from the superpixel set, like this. Based on the difference between the obstacle target and the background area, the possible obstacle superpixel in the initial boundary set are eliminated. Step E is to model the superpixel set using Gaussian distribution. The subsequent step F calculate the probability that each superpixel belongs to the background through the Gaussian distribution. Now we have obtained the background and foreground probability for each superpixel. Step G. Use an optimization framework to fuse the foreground and background probability to calculate the saliency map. The obtained picture is relatively rough, and the obstacle and the background cannot be well divided. Step H is to use a threshold to penalize the saliency map. Finally, we can get the position of the obstacle through the saliency map. This is the result of our saliency detection and obstacle detection. The first row is the original image. The picture in the second row is the result of saliency detection. And the picture in the third row is the result of obstacle detection. We can see that no matter whether there is an obstacle in the input pictures, or how the position in the obstacle change in the image, or whether there are good lighting conditions when acquiring data, the algorithm can get good results. But we also need to point out that our algorithm can't handle the reflection of obstacle in seawater well. The algorithm will also recognize the reflection as an obstacle. In particular, this can interfere with the identification of large obstacles. This can be seen from the pictures in the second column. The above is a brief introduction about the paper Real-Time Obstacle Detection based on monocular vision for unmanned surface vehicle. Thank you for your listening. The specific detail of the algorithm can be known by reading our paper.
Hello everyone. The topic of my paper is a method of data integrity check and repair in big data storage platform. I will discuss it from the following aspect. With the rapid development of information technology and the increasing popularity of the network, the amount of data and information has grown rapidly. The world has entered the era of big data. In recent years, the Internet of Things industry in China has developed. The products of the Internet of Things industry are increasing rapidly under the expansion of market demand. So the data that the Internet of Things needs to process is also very large. In a big data environment, large amounts of data are stored remotely on the big data platform and distributed across different storage nodes. In recent years, data corruption or loss on the storage platform has caused users to face great stress, causing frequent loss of events to users. In order to overcome the possible damage and loss of data on big data platform, this paper designs a secure and reliable data integrity verification and repair method based on HDFS distributed storage system, which provides an effective guarantee for the integrity of user data stored on big data platforms. Hadoop is a software framework for distributed processing of large amounts of data. Its core is HDFS, which is the basis of data storage management in distributed computing. The HDFS cluster adopts a master and slave structure mode and consists of a name node and several data nodes. It is deployed on chip machines with high fault tolerance and high scalability. In the structure, the CRC32 checksum of the data is calculated and written by the client when reading and writing data to the HDFS. HDFS calculates the checksum every fixed length, and the checksum is saved with the file block. For a large file, it takes a long time to divide it into small file blocks and then verify it. The calculation is inefficient and wastes storage resources. Since CRC32 is not considered for data security, it cannot detect whether the file block has been tampered with. The attacker can generate a fake file with the same cheat code through the original CRC32 cheat code. At this time, the file block is replaced, but the CRC32 value is still the same and cannot be detected, and the data integrity and reliability check cannot be achieved. HDFS, on the other hand, HDFS scans every 504 hours. If file block corruption occurs during this time period, the system cannot detect it, and the data integrity in the storage system cannot be guaranteed. In the HDFS system, in order to implement security checksum repair for stored file blocks, this paper designs query model, verification model, and repair model. The query model is configured to query storage information for the file blocks in the system, process user operations on the data nodes, and query requests of data nodes. The verification model is configured to periodically check the file blocks stored on the node to detect whether the file block is damaged. The repair model is used to process corrupted file blocks on the data nodes to implement file block repair. The interaction between the models is shown as a figure. When the verification model finds
Good morning, ladies and gentlemen. Thank you all for coming. My name is Fan Yi Gao. I am a graduate student from National Taiwan Normal University. This research was completed by Professor Fan Xun Zheng and me, and I am very pleased to present our research resource to you today. My English is limited. I did appreciate your patience with my accents. The subject of my presentation today is a study of image recognition for standard convolution and Depthwise separable convolution. I did like to conduct my presentation in the sequence shown on this slide. This presentation will be divided into 5 major parts. I'll start off by giving you the theoretical framework of my study. Then I'll go on to outline the research method used in this study. At last, I did like to highlight some of the significant results I have found from this study. First, introduction. The success of Google's AlphaGo main machine world in 2016 made artificial intelligence, which had been developed for more than 6 decades, once again attracted the attention of scientists, which embraced the acknowledgement. In 1998, the first model architecture used convolution neural network. However, due to the development difficulties of the hardware environment at that time, the computing speed of the computer had not yet been improved. The storage space was small. The development of AI was at the bottleneck. In 2006, Hinton proposed the deep belief network, which successfully tried multi-layer perceptron. In 2012, used graphics processing unit and coupled with a deep learning model of CNN, and then proposed an architecture called Velocities to the ImageNet computation. This paper focuses on the difference between Depthwise separable convolution and standard convolution in deep learning and the effect of using data augmentation. Second, related works. Convolution neural networks use feature engineering to extract useful image features, so it has a significant effect on image recognition. Convolution neural networks are structured by convolution layer, pooling layer, and fully connected layer. The convolution layer performs a convolution operation on the original image and a specific feature to extract a feature map. The pooling layer is connected to the convolution layer, and then down sampling is performed on the picture. It is a method to reduce the amount of data in the picture and retain important features. There are two most common methods of pooling: max pooling and average pooling. In MobileNet, a Depthwise separable convolution that can disassemble the convolution layer operation is proposed to construct a lightweight deep neural network. Compared with the standard convolution parameters, the footprint fender is very small, and the kernel does not change over time, showing competitive performance. That's why separable convolution divides the convolution into two paths for operation. Depthwise convolution and pointwise convolution. Depthwise convolution is to create a k by k kernel for each channel of the input data. Then each channel performs convolution for the corresponding convolution kernel independently. Pointwise convolution is the performs one by one kernel for each completed channel. One by one kernel help decomposition, channel feature learning, and spatial feature learning. Keras is an open source advanced deep learning library that uses minimal code and takes the least amount of time to build deep learning models. Keras is a model-level library that provides advanced modules needed by developers, but Keras only handles model creation, training, forecasting, etc. Third, proposed analysis approach. This paper is based on standard CNN and Depthwise separable convolution to build training models. The image size of the input dataset in standard CNN is 32 by 32 and performs one convolution operation. The feature is set to 3 by 3 to generate 32 sets of features followed by a maximum pooling operation. The feature is set to 2 by 2 and the size after image down sampling is 16 by 16 and there are 32 groups of 16 by 16 images. The overall architecture is shown in table. Each convolution layer uses ReLU as the convention function. A dropout layer is added to randomly discard 15% of the nodes. Finally, an output layer is established to output a total of 10 nodes corresponding to 10 categories in the dataset. After that, the softmax convention function is used for convention, which can convert the output of the nodes into the probability of predicting each image category. The first layer is a standard convolution layer. The input image size is 32 by 32. The feature is set to 3 by 3 and the 32 sets of features are generated. The batch normalization and the ReLU are enacted as convention functions. Next, a layer of 16 groups of features of Depthwise separable convolution, which a stride of 1 and a layer of 128 groups of features of Depthwise separable convolution, which a stride of 2 are enacted to generate 128 groups of 16 by 16 images, then build sequentially. The global AVG pooling layer is enacted to solve the problem of too many parameters in the fully connected layer. Finally, an output layer is established. Output a total of 10 nodes. Data augmentation is the processing of the dataset of the original image to expand the number of datasets and improve the problem of insufficient raw data. Data augmentation technology processes a new image by rotating, resizing, scaling, or change the brightness, color, temperature, footprint, and so on. Although for humans, the two images can still be identified as the same image, but for the machine, the new image is a brand new image. So the image recognition model can improve the performance to a certain degree. In addition, data augmentation technology can also prevent overfitting of the model, improve the recognition accuracy of the model, and obtain a network which stronger generalization ability. Fourth, experimental results. This paper uses CIFAR-10 dataset, 16,000 images in size 32 by 32. Among them, there are 16,000 images which a size of 32 by 32, of which 50,000 are used as the training sets and 10,000 are used as the test set. From the 50,000 training sets, another 10,000 are used as the validation sets. There are 10 categories in total, including airplane, automobile, bird, cat, deer, dog, frog, horse, sheep, and truck. There are 6,000 images in each category for a total of 16,000 images. The operating system is Windows 10, 64-bit version. The main deep learning tool is the establishment and the training of code processing models developed by Keras. The programming language environment is Python. Batch counter size for each architecture is set to 64 and the epoch number is set to 100. Standard convolution represents standard convolution neural network. Depth saver convolution represents Depthwise separable convolution. Standard convolution DA represents a standard convolution neural network led the data augmentation technology, and the depth saver convolution DA represents a Depthwise separable convolution led the data augmentation technology. Data augmentation technology is mainly targeted as training sets. The experimental results of the training concentration accuracy show that the Depthwise separable convolution augmentation can obtain a higher accuracy than the convolution. Therefore, this experimental further explored the impact of adding to architecture to data augmentation technology on model accuracy. From the experimental resource to verify the centrality accuracy of the concentration, we find that the Depthwise separable convolution and the standard convolution neural network are lower than that of the training set. However, the Depthwise separable convolution architecture is still lower than that of the standard convolution neural network. Higher accuracy can be obtained. In addition, with the addition of data augmentation technology, the accuracy of recognition resources for both architectures have improved. Experiment results can be observed that the Depthwise separable convolution architecture achieves higher accuracy than standard convolution neural network. Depthwise separable convolution architecture is composed of depthwise convolution and pointwise convolution, leads to an increase in the number of layers. Even though there are many layers of depthwise separable convolution, the number of parameters is still lower than the standard convolution. Therefore, the memory usage can be reduced during the model training process. In addition, although the addition of data augmentation techniques may lead to a decrease in the accuracy of the training set, this can improve the accuracy of the validation set and improve the generalization of the model. Finally, conclusions and feature works. The experimental results showed that although the Depthwise separable convolution architecture can reduce the number of model parameters, it will increase the number of layers in neural network. Thereby, it will not speed up models' training time. In general, computing capability is limited by hardware-based limitation. The architecture of Depthwise separable convolution is able to reduce memory usage and maintain recognition accuracy. In the future, we expect to optimize the architecture of Depthwise separable convolution, such as tap and drag feature shape and input size for pursuing the higher recognition accuracy. Thanks for your attention.
Hello everyone. The topic to be reported today is a novel gene algorithm-based DSK generation scheme. I report Mingyan Cai. And then here is outline of this presentation. Let's talk about the introduction first. Nowadays, human beings can often desire information and advice through various network mediums, such that e-commerce and the digital economy are newly developed transaction methods in recent years. Science DES has a long history. Many scholars have questioned the security of the DES. The major criteria include the insufficient length of DES and the insufficient resistance to perfect attack, especially in the era of rapid growth in hardware computing performance. Security threats have also increased rapidly. In addition, DES has the problem of weak key. If a weak key is used, an encryption key, an attacker will greatly increase the chance to crack the password. But DES is fast and convenient. Many servers still use symmetrical DES as the main encryption mechanism. Lastful, this study will also adopt the symmetrical encryption concept and design a new encryption method to respect the shortcomings of DES. Let's talk about the introduction first. What is the meta-heuristic algorithm? Meta-heuristic algorithm is a solution search method in the field of the artificial intelligence. It solves the combinatorial optimization problem in a suitable time and space. Paper use the gene algorithm is a simulation of the natural evolution of an algorithm concept from Darwin theories of the evolution. Approach GA negative mechanisms, which are solution, crossover, and mutation. GA can balance global search and local search. Based on these perspectives, this study attempts to make key generation problem to appear solution search problem. Then, let's talk about encryption based on gene algorithm. Many scholars have proposed the use of GA for Raven 3. The other use GA to generate the key. The first step must be decision of a fitness function, so that the key can be distinguished from the chromosome. Because GA is a random process, the generated key can largely avoid the possibility of weak key generation. However, the other use channel entropy as the fitness function, so that even if the process of the key generation is based on a random process, the continuously characters in the key are continuously in questions, so that channel entropy will still determine that it is a legal and secure key combination. Although the crossover and the mutation of GA can avoid this situation for legal keys, it is impossible to avoid this situation for key. They are original characters. In short, GA cannot completely change the key combination and still has a firm chance to encounter the deliberately decision attacker. Therefore, this study will not only make full use of the GA characteristic to find the best key combination, but also decision the fitness function to equal any factor that the key is maturely cracked as far as possible. Then we define the problem. The fitness function based on channel entropy cannot effectively rule out the generation of the continuous key. The so-called continuous key means that there is only slash translation relationship in the key combination, so the gap between adjacent bits is very small. We can find from Fig that 16 pieces of data were randomly obtained from the 192 pieces of data generation using GA-based encryption reference. That's the case of the continuous key combination will exist in this sample. We can find that there are serious problems in the 4 records. That's the gap of less than 4 occurs between 2 adjacent positions that are marked with the red box and the blue box. These results are easy to solve regularly and brute force attack, which are also called continuous key. In order to visualize the similarity relationship of the key, we convert the penalties and the key into the form of bytes and map them to the Euclidean space as shown in that figure. The red line is the penalties, and the black line is the generate key. The area enclosed in the median is smaller, which means that both of them have a high similarity. Therefore, to determine whether the key is good or bad as long as just calculated the size of the area formed between the two lines, the normally linear programmer model can be shown as right figure. Let's make mark as this meter is alpha ij is greater than equal to 0 and alpha ij less than equal to 255. Beta is greater than equal to 0. Gamma is greater than equal to 0. Then area S can be defined as the number 1. S is the total areas. Gamma is the number of times the panel lines and the key line. Alpha ij is the area of the line segments I to j. N is length of the key. In order to highlight the characteristics of the area and the case where there is no intersection, we define the beta variable as number 2. Line segment ij will be divided into the two areas by the reference point k. Because the two curves might have an oscillating relationship, this feature will affect the similarity judgment. So the two areas are subject to take the absolute values and divide by 137. If the line segment ij does not have an intersection point, it indicates that the two curves may not have an oscillating relationship, which indicates that the similarity is large and the value is 0. At this moment, the general solutions are their extreme value, although they will oscillate. At this moment, the general solution area and the extreme value, although they will oscillate, they are still too easy to be deciphered due to regularity. Therefore, in order to avoid the general solution as always at their extreme value, we first determine the value in the key to offset value and decide whether to accept the penalty mechanism according to the tau. We will see number 3. The random values are ranged from 0 to 10, and the 127 is the maximum value of each unit. Assuming a value greater than tau indicates that it may be closer to the extreme. If more bits of the key are closer to the extreme value, the resulting area may seem large, but it is unbelievable. Therefore, a punishment mechanism must be used. When a number approaches the extreme value, the lower the key source. We will see number 4. X is the number of occurrences where the value is greater than tau, and omega represents a penalty value between 0 and 1. This study is set as 0.88. The intention is to make the negativity response of this value greater than tau decrease exponential. The next graph is the result we expect. In this chapter, we will talking about proposed mechanism. The main process of the proposed GADS mechanism in this study is to convert the panel test into by then map to two-dimensional Euclidean space. GA is a main component for key generation next. The general key and the panel test will be converted into bits to operate XO to obtain the ciphertext. The ciphertext will be treated as the panel test and perform XO operation with the next key. This proposal will be repeated 60 times sequentially. The complete process is shown in figure. The GADS will first randomly generate 12 chromosomes as the parent population. that a chromosome has 8 genes. Each gene is a random number between -128 to 127. Every chromosome must calculate its fitness values. Then all of the chromosomes will enter the 3GA key. Step 1. Selection. 2 chromosomes will be selected from the parent population as the father and the mother for operating cross by Turner method. Selection to crossover. The chromosome is first converted into binary and a random number is selected from 0 to 62 as the intersection. Point 3. Mutation. Randomly select 10 positions for mutation. The mutation may change 0 to 1 and 1 to 0. Finally, replace the 2 chromosomes which have the worst fitness value in the parent population. And the fig is the pseudo code. In this chapter, we will show the results of the experiment. In experimental setup, we use the original DES algorithm published on GitHub. The existing work as Raven3 use GA-based encryption, which adopt the same hardware setting with this study as well as the GA parameter uniformly use 12 size parent population and execute 16 runs. Each run is executed 200 times. The selection of lesion of the GA adopt tournament selection. The crossover and the mutation trigger rate are 95% and 50%. In the case of the panel test is APPLE-32. Fig 5 to Fig 10 show the similarity of the 16 keys generated by the original DS GA-based encryption Raven3 and our proposal GA-DS. From Fig 5 and Fig 6, we can find that although there are some large arrays occur between DS curve and the panel test curve, essentially there are a few sets of the keys that still have the smaller size of areas. It means that the security distribution of the original DS is uniform, causing some keys to be safe, but others are easy to be guessed. GA-based encryption can improve security through a random process. There's still a chance to generate a continuous combination. If someone with a bad intention should understand of the GA process, it is still possible to expose the data to dangers. That it is shown in Fig 7 and Fig 8. As shown in Fig 9 and Fig 10, the areas of the GADS has a large difference. It indicates that the similarity between the keys and the keys are small, so the probability of each key being guessed is low. The areas of the GADS is also larger than the other two maps, because GADS has a high degree of variability and diversity, so that the probability of being cropped is greatly reduced. Figure 12 summarizes the area of the key combination. In order to increase the characteristic of GADS, we use the equation 4 to exclude similar features to highlight the similarity of each key. It can be seen that GADS has the large dissimilarity. The final part is convolution. The key and the panel test are limited to 64 bits, and the XOR operation is repeated 16 times, which makes the key combination more diversified. Improve the defense of the GA-based encryption. which uses general entropy to design the fitness function, lets the linear continuous keys have a large opportunity to occur. This math can increase the completeness of each key. When the sensitive keys are finally combined, it can be found that some of the same values sometimes appear, so that, although it does not cause too many problems, it also greatly reduces the diversity of the overall portfolio.
Thanks for listening.
Developing an intelligent agricultural system based on long short-term memory. Outline. Introduction. System model. Long short-term memory LSTM. Algorithm. Experimental results. Conclusion. Introducing. The key features present in this study are: 1. Reduce the electrical wiring costs by using solar power. 2. Adding weather forecast information to initiate the equipment and avoid the waste of electricity. 3. Use the environmental monitor to check whether the trip is at a suitable environmental and the system will alone if the environmental is not suitable. System model. The intelligent agricultural system based on LSTM provided by this research conducts intelligent agriculture IoT to detects the environment. Long short-term memory (LSTM) algorithm. The study implemented LSTM for soil and temperature prediction. Due to the lack of electricity and water resources in Finland, it is vital to monitor and control the intelligent agricultural system. The system factors in lead, electric server, electricity, fuel, solar power. Yet, the IoT system could not operate all the time under limited power. To avoid exhausting electricity and lead failure of initiating the IoT setting, the server is set to set weather forecast information and combine the temperature and still humanly collected by the IoT standing to conduct analysis. Inputting the Cretan formula temperature, soil humidity, and weather information to start the computation of similar. The result will be a value between 0 and 1. Afterward, the output later value will also be a number between 0 and 1. Figure 3 shows a hardware used in the system. Figure 4 demonstrates the experimental field. And figure 3, this is IoT system. Figure 5 use XMPP to combine IoT picture information. Figure 6 and Figure 7 utilize LSTM to predict farm environmental. The server uses the prediction to define commit to the IoT system, which initiates the equipment to detect farm environmental bug. The fall data will be sent back to the server for LSTM to mix real and prediction data for compression and correction. Figure 5 is the IoT platform. Figure 6 presents the LSTM environmental. And the LSTM prediction curve is shown in Figure 7. This is LSTM prediction curve. Convolution the system utilizing LSTM to predict the initiating the IoT system through the prediction to power up the system. Eventually, long electricity wisdom and transfer the safe energy for control water resources. The winter over by this article significantly reduces the intelligent test of intelligent agriculture. Thank you.
Hello everyone. The title of my article is Detection of Atherosclerotic Lesions Based on Molecular Communication. First, atherosclerosis is a very common vascular disease in humans. There are many reasons for this type of disease. For example, if the vascular dynamic environment is complex, it will cause inflammation of blood vessel damage, which will make the lipid take advantage of the endothelium. The membrane thickens, smooth muscle cell proliferation, and plaque formation. And sometimes the enhanced local permeability can also cause the deposition of lipids and lipoprotein particles in the lower layer of endothelial cells, and the oxidation of lipoproteins in the inner membrane can cause immune reactions, namely the activation of endothelial cells, monocytes, and expression of receptors in lymph cells. Cholesterol is a kind of lipid absorbed in the digestive tract. The combination of low-density lipoprotein and cholesterol will be endocytosed into the cells. If LDL cholesterol is not absorbed into the cells, the excised compound will be transported into the blood vessel wall. The increase in plasma cholesterol concentration is accompanied by increased oxidation of low-density lipoproteins, which in turn promotes the conversion of monocytes and macrophages to form cells. Their continued deposition in the intima can lead to atherosclerotic plaques or formation of lipid strips. As high cholesterol, endothelial cell damage, and abnormal hemodynamics continue to function, lipid bands graduate fibrous lipid plaques, including multiple layers of smooth muscle cells, connective tissue, macrophages, and T lymphocytes. Further growth will form fibrous caps and mature atherosclerotic plaques. As shown in this picture, from the normal state of the blood vessel to atherosclerosis to tearing, there is a process of intima hyperplasia, intima lipid deposition, media fibrosis, and media hardening. With the development of atherosclerosis, the area of the blood vessel lumen used to supply blood downstream gradually decreases. Therefore, we can map the degree of atherosclerosis development with the degree of reduction in intima cross-section area. This is my idea. In fact, in reality, there are many methods for observing blood vessel-related diseases, such as angiography, which can eliminate the influence of bones and soft tissues and other unrelated structures on the image, and can reflect the status of the same location after a period of time. The contrast agent used in medical is mapped into a nanorobot in this article. Obviously, blood flow is significantly affected by the cross-section area, the lumen of the blood vessel. The angiography technique is used to observe the difference in the concentration of the contrast agent between the diseased blood vessel and the normal blood vessel at the same location with the time intervals. That is, the difference of the concentration level of the nanorobot. due to the beating of the heart. The blood flow velocity in the blood vessel of human body is a non-stationary flow that changes approximately periodically. And due to the mechanical characteristics of the blood flow, the blood flow velocity has a certain distribution in radial direction of the blood vessel. The relationship between the detection probability of the nanorobot and the cross-section area of the lumen is caused by USEF, perhaps molecular communication noise, and capacity analysis for participate drug delivery system. Just this. The simulation tool uses LabVIEW, and the simulation results are shown in the figure, which clearly fits the attempt. That is, as the cross-sectional area of the blood vessel becomes smaller, the detected probability becomes smaller. And this result is consistent with Whitmore's curve of the relationship between the apparent viscosity and the rate of shear strain in reality of circulation. The conclusion is based on the laminar flow of the reagent channel. At the initial stage of the blockage of the blood vessel, the probability of the drug received at a fixed point is inversely proportional to the cross-section area of the blood vessel. When the blood vessel obstruction is more serious, the cross-sectional area of the blood vessel decreases, and difficulty of blood flow passing further increases. Surely, for actual human blood vessels, the elasticity of the blood vessel wall must be considered. Generally, after the atherosclerotic plaque develops to a medium or higher level, that is, 50% to 99% of blood vessel obstruction. Once the remaining part of the lumen encounters a blood clot, it will block blood flow. After a thrombus ruptures, as the blood flows to the distal branch arterioles, it can cause ischemia or necrosis of the tissue. In reality, blood vessels are elastic. With the development of the disease, the arterial vascular wall remodels. In order to adapt to the disease, and the luminal cross-sectional area does not change, the vascular wall will become thinner. However, once the limit of vascular remodeling is reached, the diseased area begins to protrude into the tube area. The cross-section area of the blood vessel decreases, and the blood flow is gradually blocked. With the increase of occlusion, cross-section area of blood vessel circulation becomes smaller and smaller, and the hemodynamics of vascular stenosis become more complicated. This article uses the commonly used medical angiography technology to model the cross-section area of the blood vessel with the idea of molecular communication, and related it to the more iconic nanorobot parameters, namely the contrast agent to measure the development of atherosclerosis. So, if the disease process can be measured with specific parameters, then the doctor's labor costs can be reduced. That's all. Thank you.
Hi, I'm Zhang Ruizi. I'm from Chengdu University of Technology. I'm very glad to attend the BICT meeting. Today, I will make a report on our research on behalf of our MIT. The title of our study is Designed for Detecting Red Blood Cell Deformation at Different Flow Velocity in Blood Vessel. Speak to blood disease. Most people's image is stuck on thrombosis, which is one of the diseases with a wide range of symptoms. Many studies have been carried out worldwide on the topic of detecting thrombosis in vivo, and many monitoring schemes for thrombosis have been proposed. We know that there is a market now for testing human cells with blood cell analyzers, which are used to detect normal cell morphology. This kind of cell detector can only detect the cell morphology in vitro, and the volume, conductance, and laser detection methods only focus on the static cell morphology, which is very lacking in the detection of cell deformation. In the human blood environment, the reduction of RBC deformability is the cause of many diseases such as hemolytic anemia, ischemic anemia. Specific disease we are familiar with, such as sickle cell anemia. It is also a direct physical expression of many different diseases such as malaria and diabetes. The ability to detect RBC deformability cannot be only direct analyze individual blood conditions, but also assist in the judge of polycythemia, malaria, and other diseases. Currently, there are many methods for the determination of RBC deformability, which can be divided into two categories. The first category uses RBC suspension to indirectly estimate and compare the average deformability of RBC population. The second is the use of a single red blood cell to determine its ability to deform and the mechanical properties of cell membranes. In this paper, we propose a molecular communication system for the rapid detection of RBC deformability in blood vessels. The system is based on nanoparticles released into the bloodstream and then measures the concentration of nanoparticles at the receiving end. Statistical measurements of the absorption process can infer the shape of the cells without resorting to slow and invasive blood tests and in vitro tests with large errors. The system can be realized by means of a device that can release the molecules in the container and monitor their downstream absorption. Health status can be distinguished from pathological status based on the statistics of the signal received. Here, we measure the deformability of red blood cells based on whether they are healthy or not. I will start with the following sections: the mathematical model, the design of the system, simulation results. First, the specific modeling process is as follows. The first is the modeling of the blood environment. This design focuses on testing in the human body. In previous studies, molecular transport in the blood vessels was generally regarded as molecular diffusion, and blood as Newtonian fluid. This paper focused on the analysis of red blood cells morphology, which is non-Newtonian fluid. And there is a modeling of the red blood cells. In order to match the characteristics of the actual red blood cells, the stress-free shape of the elastic spring network was used to model the red blood cells in the blood. And finally, the nanoparticles. Nanoparticles are modeled as rigid particles with a volume similar to that of pellets. They flow along the linear flow through blood drainage in the blood vessels and the collision with the red blood cells and the wall of the tube is the drawing force. In the presence of red blood cells, the random collision between the nanoparticles and the red blood cells will affect the speed of the nanoparticles. When the flow rate increases, each fixed flow state corresponds to different distribution probability of the cell state. When the ratio of cell size to vessel size is fixed, the red blood cells will eventually stabilize in the rolled over state, sleeper tank state, and parachute state after single dimension analysis. Different forms of red blood cells have different degrees of resistance to the nanoparticles, as shown in the figure. The obstruction is regarded as the modulation in the process of signal transmission. The red blood cells in different states are the modulation signals. The nanoparticles are the carriers, and the final concentration at the receiving end is the required information. After modeling, we design the concrete system. At the nanoscale, Brownian force acts as the dominant force driving nanoparticles to the surface of the blood vessel wall. However, the resistance acting on the nanoparticles is relatively small. In the design, the resistance of the nanoparticles mainly depends on the state of red blood cells. At different flow states, a certain concentration of nanoparticles is released into the microtube and received at the downstream. The receiving end receives and records the concentration of nanoparticles. By comparing the difference in the concentration between the transmitter and the receiver, the degree of RBC deformation was predicted, the blood state was determined, and the existence of blood disease was judged. Of course, it's a preliminary judged. The simulation shows that when the flow rate is relatively low and the red-blue cell is located in the center, the blood flow is subjected to extrinsic shear stress and begins to rotate. In this case, the fluid stress is not sufficient to convert the RBC shape to the parachute or slipper shape. In other words, at lower flow rates, if the cell is normal, the particle receiving concentration will not be affected by changes in cell shape. Further, if the concentration changes significantly at the time, it indicates that the cell is in abnormal shape, such as teardrop shape, or the normal function of the cell is lost. We all know that red blue cell death leads to increased blood viscosity. The cell deformation capacity can be determined by directly comparing the concentration of the receiving end. If in extreme cases, such as cells chlorus, the concentration at the receiving end does not change significantly, regardless of the amount of blood flow in the vessel. Those comparisons are made after the cell has reached a stable state. However, when the cell morphology is very easy to change, the concentration of the receiving end is also unrelated to the blood flow, which does not conform to the above rules and presents a random character. To verify our design, we performed the following simulation. In the simulation, we first draw a probability curve about the observed state of RBC. For fixed size RBC, we use shear rate as the main parameter to control the shape of RBC. Therefore, we assume that the change in shear rate in the figure also represents the corresponding change in the shear modulus of cells. In the previous inference, we knew that the velocity of the nanoparticles was related to the velocity of blood flow. Moreover, if the time is long enough, the small change in blood vessel caused by RBC deformation is ignored, and the velocity at the receiving end of the nanoparticles is not affected by RBC deformation. Just as the figure shows, the concentration of nanoparticles in the blood vessel changes over time, as shown in the figure. And next, the three curves with different flow rates indicates that the concentration of nanoparticles was hindered to different degrees at different flow rates. The smaller the flow rate was, the greater the degree of obstruction would be. On the contrary, the larger the flow rate was, the smaller the degree of obstruction would be. The above is a brief introduction to our simulation. Finally, in this paper, we propose a solution for the detection of RBC deformability based on the effect of RBC deformability on molecular transmission in blood vessels. Combined with the existing experimental data, the feasibility of detecting the deformability of a single red-blue cell was demonstrated through specific activity analysis. More specifically, given the molecules released into a blood vessel, the simulation showed that the abnormal cells affected the number of nanoparticles the receiver could receive compared with the normal conditions. It can also be seen from the results that the distribution of nanoparticles in the blood vessel after reaching steady state does not depend on the blood flow velocity. So, the design is applicable to all blood vessel models with different flow rates. The detection of cell deformation can also be detected by its light scattering properties. The transmitting end emits light of a certain wavelength, which is absorbed by the receiving end and compared with the transmitting end to predict the cell shape. The results of this paper can be further expanded to carry out more probes. For example, the aggregation effect of multiple red blood cells at low flow rate corresponds to deformation effect at high flow rate, further enhancing the design applicability.
Good afternoon, everyone. I am Peng He to give a talk here. Today, the title of the presentation is Intelligent Power Controller of Wireless Body Area Networks based on Deep Reinforcement Learning. We have 7 co-authors. They are 刘真丽、傅雷、陶中元、刘佳、唐彤 and 李直度. All of the co-authors come from Chongqing University of Posts and Telecommunications. This talk consists of 5 parts. At first, it introduces the technical background. Then, I will present the basic issue, deep reinforcement learning, which supports the power controller design in the next session. After that is the power controller design. In this part, we propose a model to optimize the performance. In the simulation part, we present how deep reinforcement learning is proceeded in process of power controlling. It checks the performance of proposed power controlling scheme. In the finally, I will give a summary for this work. In recent years, W-band plays an increasing important role in the applications of medical monitoring and treatment. A typical W-band is a 3-layer architecture, which is composed of several distinct nodes. Some important ones are sensor terminals, which collect psychology data from different biology medium, like EEG, ECG sensors. Another important node is the W-band coordinator, which is named center node in this work. It receives, processes, and forwards the psychology data to the data center. The rapid progress of wireless communication technology has promoted the progress of wireless body area networks. For example, the distribution of 5G technologies improves the capability of data transmission and enables more potential applications. In this work, we study the power controlling issue for W-band. Sensor terminals are limited by their size and structure. Thereby, they have limited energy. In addition, the charge costs of the sensors are high, and sensors are also difficult to be replaced. Thereby, how to save energy is an important issue for W-band, and power controlling of the nodes are an available way to realize that goal. Now, I will briefly introduce deep reinforcement learning. Deep reinforcement learning is one class of machine learning which applies the neural structure of deep learning and the interactions between machine and the environment. In W-band, the sensor node collects biological information from the body, interacts with the coordinator, deploy machine learning algorithms in computation, and finally take action via power controlling. In this work, we adopt 4-layer neural structure. The first layer is input layer. The second and third layer are two middle layers, and the last layer is the output layer. There are 5 major elements. The first is the status. They are denoted by ST. The second is the actions. They are denoted by AT. The third are the places. They are denoted by PI. And the fourth are the rewards. They are denoted by RST, AT. The fifth are strategies. They are denoted by QPI, ST, AT, theta. In the following, I will introduce how power controller is designed. As shown as this figure, the system we considered are composed of 2 W-bands. Each W-band is assumed distributed for one person, composed of several sensor nodes, one central node, and the collection with remote data centers. We consider the interference between different W-bands. The received signal of the central node is composed of 3 parts, which are spreading signals from itself W-band, the spreading signal from the neighboring W-band, and the noise. Here, XT denotes the transmitting signal from the sensor node, which is related with the transmitting power, P. HT is the response function of the channel. The signal to interference plus noise rate is denoted by their power ratio. Here, the capital H is the Fourier transforming form of lowercase ht. The capital N is the power of white Gaussian noise. The channel capacity is expressed by Shannon formula. To proceed the power controller of the central nodes, we optimize two targets. The first, we maximize the channel capacity U. Second, we minimize the transmitted power P. The optimization is preceded by the neural structure, in which the learning process relies on the reward function, which is designed as a logarithm of some ratio of channel capacity and transmitting power. In the next session, we will introduce the simulation parts of this work. We will first introduce how the simulation is designed or considered, and then we present the numeric results and do some analysis. We apply the MATLAB 2019A as the simulator to validate our work. In special, we use the AI toolbox of MATLAB as the base to realize deep Q-learning and incorporating the design of power controller. Major parameters are as follows. Transmission power is located between minus 50 to 50 dBm. Noise power is set as minus 100 dBm. Communication bandwidth is set as 10 kHz, and the number of nodes in one W-band is located between 5 to 15. The discount factor of the DQN is set as 0.9, and the learning rate is set as 0.5. This figure presents the accuracy performance of DQNPC in 4 epochs. The blue 4 line is the training accuracy, and the black dotted line is the validation accuracy. It can be clearly seen that training accuracy has some fluctuation with the increase of epochs, and finally settles in 1% in the third epoch. Differently, validation accuracy increases smoothly and settles in 86.67%. The results indicate that our proposed DQNPC is feasible and with no complexity. This figure shows how energy consumes in transmission of unit bits for each node. The result reveals that applying FPC will need the rapid increase of energy consumption for W bands, since increase of nodes results in heavier interference, and nodes may spend more time and energy for successful communication. However, proposed DQ-MPC is more intelligent to dynamically elevate the inter-network interference. As a result, the energy consumptions decrease. This figure shows the relation between average node throughput and the number of nodes per W band. It can be seen that node throughput decrease with increase of node number due to the increased interference. By comparing the performance of two algorithms, we can see that throughput of FPC is a little higher than that of DQ-MPC. Such difference of nodes throughput decreases with the number of nodes and become inconspicuous when node number is large. We infer that DQ-MPC increases the performance of energy consumption by sacrificing limited performance of throughput. In the next, we will give a summary for this work. In this work, we studied the dynamic power controlling scheme, and we proposed a deep Q network-based power controller, and we verified the performance of proposed DQ-MPC. In the future, we will study the performance of the DQ-MPC in more complex scheme of W bands, and study algorithms based on the structure of DQ-MPC. That's all. Thanks for your attention.